Hello everyone, I am Swamideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Medtronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Soumyadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as an area sales manager in India Medtronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Soumyadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as an area sales manager in India Medtronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now, I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Medtronic. I take care of the eastern part of our region.
My name is Ankita Sarkar, and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now, I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Swamindeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Swamindeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant
everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. So right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I'm Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar, Department of Chemistry, to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. So right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar, at Department of Chemistry, to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as an area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as an area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as an area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as an area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as an area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as an area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as an area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Good afternoon, everyone. On behalf of NSHM Knowledge Campus, Kolkata, I welcome all of you to the Professor Emil Schroff Hall. Our session will start in a short while. I also welcome Professor Svante Winberg, Professor of Neuroendocrinology, Department of Neuroscience, Uppsala University. Welcome, sir. Please wait for a short while. We will be starting our session within maximum a span of 15 minutes. Thank you, sir. With great pleasure, I, Dr. Amrita Chakraborty, on behalf of NSHN Knowledge Campus, would like to welcome all the delegates, guests, and participants to the eNatCon 2021. The theme of the conference for this year is Research Advancement Resilience in the Pandemic Era, a drive for innovative transformation. I am also delighted to apprise you that NSHM Knowledge Campus is celebrating its 25th anniversary this year. And to mark this occasion, this international conference is being conducted on a virtual platform owing to the present pandemic situation. I'm a bit sorry for the technical delay. Now we are going to start the session again. With great pleasure, I, Dr. Amrita Chakraborty, on behalf of NSHN Knowledge Campus, would like to welcome all the delegates, guests, and participants to the eNatCon PH 2021. The theme of the conference, Research Advancement Resilience in the Pandemic Era, a drive for innovative transformation. I am also delighted to apprise you that NSHN Knowledge Campus is celebrating its 25th anniversary this year, and to mark this occasion, the international conference is being conducted on a virtual platform owing to the present pandemic situation. eNatCon PH 2021 has enabled the privilege of knowledge sharing between eminent speakers and experts throughout the world. We hope this conference will mark the beginning of the digital era, which has emerged as an indispensable part of today's world. The crux of this conference will be the strategies to overcome the challenges that our world is currently facing after the outbreak of the COVID-19 pandemic. We expect that this conference will help all the participants to expand their knowledge, and we welcome all of you with a promise to provide an enriching experience during the tenure of the conference. The take-home memories will surely strengthen our common faith, commitment through sharing questions, answers, smiles, and empathy. Thank you very much. On this momentous occasion, it is our privilege and fortune to welcome Dr. Vante Winberg, Professor of Neuroendocrinology, Department of Neuroscience, Uppsala University, Sweden, as a keynote speaker for the upcoming session. Welcome, sir. His session will be moderated by one of our esteemed faculty members, Dr. Shobhik Roy, Associate Professor, NSHM School of Health Sciences. Dr. Roy has received his doctoral degree from Jadavpur University, India, and he is the recipient of the prestigious Matsume Postdoctoral Fellowship. Dr. Roy has a prolific teaching career spanning over 15 years, and his research area focuses mainly on targeted novel chemotherapeutic approaches. Now, I would take the pleasure to welcome Dr. Roy online.
Thank you, Dr. Chakraborty, for the kind information and introduction. Good afternoon to all the delegates, guests, and the participants. I, Dr. Shobhik Roy, have the pleasure of introducing our keynote speaker, Professor Vante Winberg. Dr. Winberg is currently working as a professor of neuroendocrinology in the Department of Neuroscience, Uppsala University, Sweden. He has received his doctoral degree in comparative physiology from Uppsala University and has carried out his postdoctoral research at the Fernald Lab, Department of Physiology, Stanford University, California. His academic experience covers a span of more than 25 years, and in this period, Dr. Winberg has published more than 100 articles in peer-reviewed journals. He has also authored more than 10 reviews and book chapters. His research interest includes fish behavioral neuroendocrinology, especially intraspecific behavioral differences and neuroendocrine mechanism mediating different behavioral profiles. His research projects are mainly focused on the fish welfare, fish anesthetics, aggressive behavior, and the stress physiology. With these kind words, I would like to invite Professor Vante Winberg to deliver his insightful lectures on the topic Fish as a Model in Biomedical and Biological Research. Thank you. So, sir, please.
Thank you very much. Thank you for inviting me to this conference and to this very kind introduction. I'm really honored. I hope you can hear me well. And I will try to share my screen now so you can see my, let's see where it is. No. So I hope you can see my presentation and you can hear me.
Yes, sir. You can hear.
Thank you very much. So my name is Vante Winberg and I've been working with fish for many, many years. And I'm going to tell you if we look at what I'm going to talk about. So my talk is going to be about why are we using fish as a model in biomedical research? And how valid is the fish as a model for humans? I mean, how similar are we to the fish? Can we really get knowledge from the fish that can be applicable to medical research. I'll tell you a little bit about zebrafish biology. What kind of animal is this zebrafish? And also then, of course, why zebrafish is becoming the most important model organism in biomedical research, because I think it is. I'll tell you a little bit about fish brain. How is it organized? What are the similarities, differences to the human brain, and a little bit about how we study behavior in fish. And finally, I will tell you a little bit about the ongoing research in my lab, and then we'll come to some summary and conclusions. So if we start with this then, why fish? And as you heard from the introduction, I have been working on fish my whole career. And for me, it's very obvious because I started out as a sport fisherman. So I've always been very, very interested in fish. And I'm a biologist by training. I'm a comparative physiologist. I've also worked a lot on, together with the aquaculture industry, especially in Norway, trying to optimize fish rearing. But nowadays, fish is becoming a very important model organism in biomedical research and biological research, and that's what we are working on mainly at the moment. So for me, it's very obvious that it was fish to work on, and I think for a lot of people, fish is becoming very important now in research. So if we first then look at the vertebrates, then you can notice that fish is about half of all vertebrates we have. Mammals, the group where we belong, is this small part. So mammals is actually a very small group of vertebrates. Fish is about half of all living vertebrates. So if there is such a thing as a typical vertebrate, it's not a mammal, it has to be a fish. And when we talk about fish, at least when I talk about fish today, I mean the teleosts, the bony fish, which is the largest group of fish. And it's without comparison the largest group of all vertebrates, as I just showed you. So within the vertebrate subphylum, half of the species are teleost fish. So today, more than 30,000 species of teleost fish have been described, and there are still several more species to describe. So we have more than 30,000 species of teleost fish. And they colonize almost any environment you can think of on the earth. We have fish living at the deepest parts of the oceans. We have fish living in small creeks high up in the mountains. All aquatic environments are colonized by fish species. So it's a very widely spread group of very diverse. And if we look at the phylogeny, I don't know how familiar you are with the vertebrate phylogeny, but if we look at it, we have the teleosts, the bony fish up here. We have the lobe fin fish over here, and that's where we belong to this group. Mammals are coming up here eventually. So over here we have the teleost fish, and if we trace it back here to this red square down here, there we have a common ancestor. So we have a common ancestor with the teleost fish, and that common ancestor lived about 400 million years ago. Here we can see this more clearly. So about 400 million years ago, we had a common ancestor, and then it diverged. So if you follow it in this direction, we end up with the mammals and humans. And if we go in that direction, we end up with ray-finned fish and teleosts, including the zebrafish. So we are both vertebrates. And since we are both vertebrates, the general organization of our bodies and how we work is very, very similar, actually. Even though it's 400 million years ago since we had a common ancestor, we still share about 70% of genetic identity. So fish and humans are much more alike than people in general believe. And if we look at the brain, which I've always been my main interest, the brain in the teleost fish, this is a zebrafish, and the human is organized in a very, very similar way. You find all the different brain parts from the human brain also in the fish brain. What is different is the size of these different areas of the brain, but we they are organized in exactly the same way. We have the same neurotransmitter systems, and it functions in the same way. So that is one of the reasons that we can use zebrafish in neuroscience. So zebrafish then, what kind of animal is it? It's a freshwater fish. You have it in India, actually. I have a colleague who made an expedition to India to get me wild zebrafish. So I have wild zebrafish from India in my lab. They are a freshwater fish. They belong to a family of freshwater fish, which is Cyprinidae, which is the largest family of freshwater fish. So it's distributed throughout Southeast Asia. Quite common, it occurs both in larger streams, small creeks, ponds, very different environments. And as I said, it's becoming one of the absolutely most important vertebrate model organisms. Why is it becoming so important as a model organism? One reason is the size. I mean, zebrafish are small. And that is, it could be a disadvantage also, but it's an advantage when it comes to lab space. You can keep zebrafish in large numbers in relatively small tanks, which means that keeping zebrafish is much cheaper than keeping rodents. I don't know the system in India, how it works, but at least in Sweden and Europe, the researchers, we have to pay for our square meters of lab that we use. So if we can minimize space, that means that we can do more research because it's cheaper. And that is one of the big reasons for using zebrafish. Also, it was the developmental biologists who started to use zebrafish. And one major reason for that is that it's very easy to get fertilized eggs. They spawn all year around, and females can spawn every 2 to 3 days. And it's very easy because before you leave the lab in the evening, you put together some male and female fish in a tank. They spend the night together, and when the light goes on in the morning, they spawn. So when the researcher arrives in the lab in the morning, they have newly fertilized eggs. So it's very convenient if you're a developmental biologist. They have a short generation time, so you can get sexually mature fish within 3 to 4 months, actually even shorter than that. It depends on how hardworking PhD students you have, how frequently they feed the fish, because the more frequently you feed the fish, the faster they grow and the earlier they become sexually mature. And as you probably know, they have a fully sequenced genome. There's a lot of animals who has a fully sequenced genome nowadays, but the zebrafish genome is very well annotated, and we have access to all these new molecular techniques. We can easily make transgenic fish using CRISPR-Cas and other techniques. And nowadays, it's not only developmental biology. The interest in zebrafish is increasing in all different fields of biological and biomedical research, including behavior and neuroscience. And if we look at zebrafish development, one important thing again for development biologists is that the larvae are transparent. So you can follow the development of the organs within the fish, and you can mark different genes, and you can put in these fluorescent proteins, and you can follow how different systems in the fish develops in the living fish, which is very convenient sometimes. So if we look at how much zebrafish is used, this is a search that I made on Web of Science. It's about a year ago now. But as you can see, it started to take off in the early 90s. Then there started to occur a lot of publications on using zebrafish as the model. And then it has just increased. And there is no tendency to this curve to flatten off. It seems to go on for at least for some time more. And if we look at, I think the last year in this graph is 2019, and then we had 4,000 publications just in that year on zebrafish. So zebrafish is really becoming very, very important in biomedical research. And at least in Sweden, zebrafish has now passed rats. So zebrafish is more used than rats as laboratory animals. There are still more mice than zebrafish used, but I think in a very short time, zebrafish will pass even the mice. So within soon, I think zebrafish will become the absolutely most important laboratory animal. So what about fish then? Some people think that fish are very stupid animals. They can't learn anything. They don't have any complicated behavior. They are more like machines doing things. But that's absolutely not true. I have worked on fish all my life, and I can assure you that fish are not at all more stupid than any other vertebrate. They have learning abilities. They have complexity similar to other terrestrial vertebrates. They're capable of motor, emotional, spatial learning. The neural substrate in fish appears very similar to the neural substrates in other vertebrates. The telencephalon has been a problem, I will show you soon, because it develops in a different way in fish compared to terrestrial vertebrates. But nowadays we have found homologies of, for instance, the mammalian hippocampus, amygdala, and other brain areas when we have identified them in the fish, so we know where they are. And this is how a fish brain looks. It looks actually very different in different species, but this is some kind of a general fish brain, very similar to how the zebrafish brain looks. And you can see that you have all the different brain parts here as you find in the human brain or other mammalian brains. You have a telencephalon, the optic tectum, which is quite big in fish where vision is important, such as in zebrafish. You have a cerebellum. You have a hypothalamus underneath there. You have all the cranial nerves. I mean, it's the same general pattern as in a terrestrial vertebrate. And if we do then a comparative neuroanatomy, if you compare the fish brain to the brains of other vertebrate groups, like the amphibians, birds and reptiles, and humans as mammals, and then you can see that all the different brain parts that we find in the human brain, in the mammalian brain, they're also present in the fish brain. What varies is the size of these different brain regions. So for instance, the most obvious difference is that in the human brain, telencephalon, the cerebral cortex, has become huge compared to what it is in other vertebrates. So the telencephalon in the fish brain is a very small part of the brain, but in the human brain, the telencephalon, the cerebral cortex, is huge, and it covers most of the rest of the brain. But if you take away the cerebral cortex from this mammalian brain, what is underneath is very similar to the fish brain. As I said, it has been very difficult to identify different areas in the teleost in the fish telencephalon. And that is because of the development. You're probably familiar with how the mammalian forebrain, mammalian telencephalon develops. It's through this process of evagination. You start with a neural tube, and then you have this swelling, the evagination of the forebrain. So it ends up like in two hemispheres like this. And you have the different areas here that will end up as different parts of the telencephalon, hippocampus, for instance. In the fish brain, the development, or in the teleost brain, I should say, because it varies between fish groups, but in the teleost brains, there is not an evagination, there is instead this process of eversion. So cells actually move out like this on the sides. And this is the final telencephalon on the fish. And as you can see, it's a very strange arrangement because you get the ventricle on the outside of the brain. I mean, in our brain, we have the ventricles inside the telencephalon. In the fish brain, in the fish forebrain, the ventricle is on the outside. And this means that these original parts, areas that would develop into different brain structures in the neural tube, they end up in very different positions in the fish brain compared to the mammalian brain. And this has caused a lot of confusion. I think we have solved most of this now. So we have identified the areas homologous to the mammalian brain in the teleost forebrain. And as I said, they have all these areas. It's just the size of them that varies. Behavioral assays then, how to study behavior in a fish. And now I'll talk about zebrafish specifically. One kind of behavioral assays that are very frequently used is behavioral assays to quantify what we call anxiety-related behaviors. As a biologist, I usually prefer to call it boldness, but in medical literature, they tend to use anxiety. So we can call it anxiety-related behaviors. A very common behavioral assay is this called the novel tank diving test. And this is a narrow, quite deep tank, plastic tank that you put in one zebrafish into this tank. Of course, this is a novel environment for the fish, so they get scared, they go down to the bottom, and they stay at the bottom. However, the fish is curious and they want to explore this novel environment. So after a while, they start to move upwards to explore the upper parts of the tank. And then how fast they move up to the upper parts depends on how bold or how anxious the fish is. The more anxious or the more shy they are, the longer they stay at the bottom. It takes longer for them to reach the top zone. And then you have a video camera connected to a computer, and you can then track the fish and get the distance moved and how long it takes for them to reach the upper part and how long time they spend in different parts of the tanks and so on. And this is a very often used test to screen pharmaceutical compounds, toxicants, and so on. Another test which also tests anxiety and boldness is what we call this scototaxis, and it is based on the fact that if you put the zebrafish in here, they prefer the dark part of the tank. So this is a light dark test. And then, of course, they go into the dark, and again, but they are curious. They want to explore this unfamiliar environment, and then they move out into the light part. And again, you film and you take the time until they reach the light part. And we can also use what is known as the classical open field test that I'm sure that you have seen because it's very frequently used in rodents. You put the fish in here and they will start to explore, but they will stay close to the walls because that's a safer area. But then eventually they will come out and explore the center of the open field. And the sooner they come out there, the bolder they are, the less anxious they are. So these are typical tests that we use to screen for anxiety boldness. And we can test them with, if we, for instance, treat the fish with SSRI like fluoxetine, they go much faster up to the upper part of this tank than the control fish. So they are less anxious. If on the opposite, they have an alarm pheromone in the skin, actually, the zebrafish. So if they release that, which happens if they are attacked by a predator, for instance, then the whole shoal of zebrafish gets very anxious and they go to bottom, which you can see here. And you can test different compounds. So you have anxiolytic compounds, like if you chronic fluoxetine, as I just showed you. Ethanol works very similar in the fish as it does in us, actually. Chronic morphine also has an anxiolytic effect in the zebrafish, making the fish move more in the upper parts of the tank. And you have anxiogenic compounds like caffeine, the alarm pheromone that I just talked about, or you can have repeated morphine treatments that then you have this withdrawal effect and the fish becomes more anxious. So this is a very validated test to use, and it's very often used to screen pharmacological compounds and also toxicants and other xenobiotics. We have actually in my lab designed a new test. We just published this, which is a multi-concentric square field test. It has been used for rodents before, but now we have developed it for zebrafish. And the reason we developed this test is because we can get much more behavioral information from the fish. What you usually have to do otherwise is to have repeated tests, testing for different kind of behaviors in the fish. And then you have to move the fish between different tests. And that is stressful, and that will affect your results. And the order in which you perform these tests will also affect the results you get. But in this test, you can test for many different behaviors in the same arena. So if we look at the arena, you have different areas here. We have a central circle here, which is then like the open field test. So how much the fish spends in the center of the tank, which is considered to be the more risky part. We also have a ramp here, which is very shallow water up here. And shallow water is also very risky for a fish. So we can see how much the fish is moving up here and how much time they spend here. And we have a shelter up here. So in this corner, it's covered by plastic. So it's like a roof. So the camera can still see the fish because we use infrared light, and infrared light past this plastic. But the fish experience this as a shelter, as a roof. So they swim in and stand under this roof. So using this multi-square field, and we have a corridor also, which is quite safe, where the fish can swim around here. So we can get much more information, and we can screen the fish for using different pharmaceuticals, xenobiotics, or whatever, and see how that affects the behavior of the fish in a much more detailed way. We are also looking at aggression in zebrafish. I don't know if you have seen fish fighting. I have a short video clip here. These are two zebrafish, and here you clearly can see aggressive behavior in the fish. So one of the fish here, this is a pair of fish, is chasing the other one, attacking, and they can actually be very, very aggressive. So if we keep them together in pairs for too long, they will actually, one of these fish will kill the other fish. And they're usually killed by the stress that they experience in this situation. So they are very aggressive fish, but it depends on the density. So if you keep them at low density, especially in pairs, they become very aggressive. If you keep them at higher densities, you don't see that much aggression. But testing for aggressive behavior could be quite complicated. It's not as simple as you think. And there are different ways to do this. I mean, what you just saw is like the bottom here, where we actually put them in isolation. So we have one zebrafish on each side, and we have a partition like this. And then we leave the fish like this, at least sometimes for a day or at least some hours, because then these fish will experience this area as their territory, and they will defend this if there is someone else coming into this part of the tank. And then we remove this partition, and the fish will come together in a pair and start to fight. And we can then easily see who is winning, who is becoming the socially dominant animal, who is becoming the socially subordinate animal. And then sometimes we keep them together, but as I said, they tend to kill each other, so they get hurt. So we don't want to do that for a very long time because of fish welfare reasons, and because of we don't want to lose fish. So then we can put back this partition. And if we have a transparent partition, they will still be able to see each other, which means that the subordinate, the one that lost these fights, will still experience this very stressful, because they will see the dominant fish on the other side. And then we can put them together again, usually once a day, and have them to fight if we want to have a chronic situation. Another way to do this is to have them fight against each other with a transparent partition present. Or actually in zebrafish, we can use mirrors, because they would fight their own mirror image. And then we also use this kind of tank. So we have a mirror on each side here, and the fish would fight their mirror image. But it's not actually the same thing, because if you fight your mirror image, the mirror image will always do the same behavior as yourself. So the fish will never experience another individual performing submissive behavior. They will always perform dominant behavior. So if we look in the brain of the fish, which we usually do, we have very different effects if they are fighting like this, or if they are fighting their mirror image. So what about learning then? I don't think zebrafish read books, but they definitely have capacity for learning. They are not at all more stupid than other vertebrates, as I told you. And this is also something that we study in the lab in different ways. This is operant conditioning box actually made for zebrafish. I don't know if you're familiar with Skinner and Skinner boxes. This is like a Skinner box that you usually use for rodents. Then, so you can have positive or negative reinforcement in this box, and you can have the fish to learn. The easiest thing to do is to have a negative reinforcement here. So you have, then in that case, you have 4 lanes in this box, and you have an electrode on each side. So you can give an electric shock. And underneath there, you have like an iPad, so you can give different colors. And I have a small video again here. So in this case, there you can see there are 4 fish here. And in this case, all of the bottom of this got green, and at that moment the fish got a small electric shock. And then we test the fish by having half of the bottom area green and half blue. And you will see that the fish very efficiently avoid the green part because that's where they got the shock. So they learn this very, very quickly. You can also study behavior in larvae. Being a behavioral biologist, I don't really know how much behavior you can see here, but you can do it in 96 well plates, and then you have 96 larvae, 1 in each of these wells, and put it into this kind of testing apparatus. So in here you have a video camera and you have infrared light, and you can measure the swimming activity of all these 96 larvae at the same time. And then you can add pharmaceutical compounds, toxicants, or whatever to these different wells. And you can see then what effects you have on the behavior of the fish. You can, of course, we use this in my lab too. We usually have larger, so we have 24 well plates or 12 well plates, because then you get more like an open field arena, so you can get much more behavior from the larvae than in these really small wells, which I think is more interesting. So this was something general about how zebrafish is used and why we can use zebrafish as a model organism to try to understand human neurobiology, human behavior. And we have zebrafish models for different diseases like Parkinson, for instance, Alzheimer, and other kinds of diseases that we want to understand and develop pharmaceuticals for. In my lab, these are 3 of the projects that we are working on at the moment. As was said in the introduction, I have for a very long time been interested in why individuals of the same species behave differently. What we in humans call personalities. And we do that in zebrafish. That's where we use our wild zebrafish that was actually caught in India. So we have selected these fish for different personality types. I would show you soon. We are also just now actually starting up a collaboration with the Karolinska Institute in Stockholm here in Sweden, trying to use zebrafish as a model to study depression. Especially they are interested in relationship between depression and disturbance of the immune system in humans. We're going to start that next week, actually. And also we have a large project going on, which is a 3R project. I don't know if you are familiar with 3R. That is that we should replace, reduce, refine. So we should replace laboratory animals if possible. We should reduce the use of laboratory animals if possible. And when we use laboratory animals, we should refine the methods. The animal should have as good conditions as possible to give us as good data as possible, and so on. So this is to optimize zebrafish rearing, how to keep them in the lab, to optimize the welfare of the zebrafish. I'm not going to talk about this because this is my PhD student, Oli Sensarma, who will present her PhD project tomorrow, I think, in this conference. So we'll hear about that tomorrow, not from me. So if we start then with behavioral phenotypes of what is known as personalities in humans. And you can look at this cartoon and see that these different kinds of behaviors. And this is something you may not think that you have a personality in fish, but I mean, maybe you should not use the word personality, but you have definitely different behavioral phenotypes. You have different types of animals within this species and within all species, actually. It's been studied a lot in vertebrates, but it's now becoming clear that we even have it in invertebrates, like in some insects, in crustaceans. So this seems to be something very evolutionary conserved. It's present in lots of different animals, that you have these two types of animals. You think that behavior should be flexible, so you should be able to respond adaptively to any kind of environmental challenge that you get. You should respond in an adaptive way. This is not really true, because the dynamic range of behavior is limited. So it depends on what kind of animal you are, how you respond to this challenge. So behavioral traits are correlated, and individuals differ in behavioral repertoire. So you have some animals are more aggressive, and they are also usually more going in, they're bolder. They like to explore unfamiliar environments. They learn rapidly, but they learn a routine and are less plastic in their behavior. The other type of animal is non-aggressive, but they're more shy, so they're more afraid of exploring unfamiliar environments. They take a little bit longer to learn, but they're much more flexible in the behavior, so they can change their behavior if the environment is changing. So that is what we mean by this personality. And I'll just show you, this is a published study. We published it 2011, so it's quite old now, 10 years ago. What we did here, one of my PhD students at the time, she did was that we had zebrafish, we weighed them, measured them, tagged them so we could differentiate between individuals, and then we kept them in isolation for 1 week. And that was just to make them forget so much as possible about their previous experience. And then we ran like a personality test. So we had 3 different behavioral tests. We had an open field, like I showed you in the beginning, to screen for boldness. We also had gave them a roof, a shelter, so we could see how long time they spend under this shelter. And we introduced a novel object to see if the fish were bold, if they wanted to explore this novel object, or if they got afraid of the novel object. So all the fish went through all these 3 behavioral tests. And then we isolated them again for a week. And then we put them together in pairs like this. So we paired fish and had them to fight, and observed who was winning and who was losing. So we got the dominant fish, a subordinate fish, and all these different pairs. And then to really got the extreme, we took the dominant fish from each pair and put them together, and the subordinates and put them together. And again, we got a dominant and a subordinate fish. And in that way, we got the really extreme dominant or extreme subordinate fish here. And what we did after that was to use this multivariate statistic model. So based on the behavior in the personality screening, we calculated a social status for the fish. And on this axis, on the y-axis here, you have the actual observed social rank of the fish. So 1 is dominant and 0 is subordinate. And as you can see, based only on the behavior the fish performed in this behavioral test, we were able to tell who was going to become dominant and subordinate. In all cases, there was no overlap here. So the behavior in this personality screening was enough for us to tell who is going to become dominant and who's going to become subordinate. And this clearly shows that zebrafish has what we would call a personality. So what we do now in the lab is that we have the wild zebrafish from India, and we screen them for their behavior, and then we take the extreme parents from each generation. And we are now in the 3rd generation, so we have created 2 lines with different personalities. And we are now, of course, interested in the genes that are controlling these different behavioral phenotypes. And we are also interested in how much the environment is modifying this behavioral phenotype, because we know that there is an interaction between genes and environment. So we want to see if we put these fish in different environments, if we, for instance, put them in environments so they become socially dominant or socially subordinate, how much would that affect the behavioral phenotype of the fish? And of course, we also look into the brain, to the monoaminergic system, especially serotonergic system is interesting here. And then modeling depression then, how can we do that in zebrafish? Again, we have fish fighting. As I just described to you, we isolate the fish, we have them to fight, and we get a dominant fish and a subordinate fish. And this is actually, the subordinate fish shows a behavior which is very similar to what a depressed person is showing, or if you use rodents, you generate what we call learned helplessness, which is a model for depression used in rodents. And these fish are very, very similar actually. And these fish, they are exposed to a chronic stress. There will be most fighting when they determine who's going to be dominant or subordinate. But even after that, the dominant fish would be a threat to the subordinate fish. So this fish is constantly exposed to a stress. And that stress is very unpredictable, because they can't tell when the dominant fish is going to bite them. It can happen whenever. And there is a loss of control, which is also a very important factor here. They have no control over their situation. It's the dominant fish here who is controlling everything. So if you look at the behavior of these subordinate fish, of course, they show an inhibition of aggressive behavior. They also show a suppression of food intake and lowered locomotor activity. And overall, whatever you look at, they have a behavioral inhibition. They don't respond to stimuli from the environment, which is again very similar to the symptoms of clinical depression. And if we look at the neurotransmitter systems, and I told you before that they are very well conserved evolutionary, so they look very similar in a mammalian brain. Here you have a brain from a rodent, and here you have a zebrafish brain. And they look very, very similar. So here you have, for instance, the dopaminergic system, you have the serotonergic system, and this is the cholinergic system. We have worked especially on the serotonergic system here, and you can see that you have the Rafa nuclei back in the hindbrain here, and it's exactly the same in the zebrafish. And the transmitters also have very similar functions in the zebrafish and in the mammalian brain. And what we observed and we have worked with for many, many years is that in the subordinate fish, we get a clear disturbance of the serotonergic system. So these are fish fighting, and these on this axis is days, and on that axis you have the turnover of serotonin in the brain of the fish. So the ratio between the metabolite, 5-hydroxyindole acetic acid, and serotonin. And as you can see, there is a very dramatic increase in serotonergic turnover in the subordinate fish, and it stays elevated. This is after 21 days of social interaction. So we think this is a very good model for studying depression, and that's actually what we're going to do in this new project now. So then if we come to summary and conclusions, so zebrafish is rapidly becoming the most important vertebrate model organism in biomedical research. And I think, at least in Sweden, it will pass mice quite soon. It started out as a model in developmental biology, but the use of zebrafish in behavioral neuroscience and pharmacology is very rapidly increasing. However, there are also other fish species, so you should not forget that there are 30,000 species of teleost fish. As an old fish researcher, I tried to convince my university that they should have facilities to keep also other fish. And I was very happy last week. I visited the Max Planck Institute down in Munich, Germany. They had huge facilities for zebrafish. The zebrafish is very, very large there. But it made me very happy to see that they also kept other fish species. For instance, in the huge facilities where we were, they also had cichlids and they had killifish. Cichlids are fish which have a very interesting social behavior and can be used for many different studies. And killifish are also interesting because they have parthogenesis, so the egg develops without fertilization, so you can generate clones of fish, which are very interesting for many questions. So this is something you should always remember, that what determines what model organism you should use in your research should be your biological question. I mean, you should not go in there and just use a model organism because it's popular. You have to think about what are your questions, what do you want to study, and then try to choose the model organism which has the best chance to answer this question. And then finally, I will just thank, of course, my lab members. There has been some changes, but still, this is Olis and Sarma, who comes from your college, who is now a PhD student in my lab. She will talk tomorrow. Johanna Axling, who is also a PhD student in my lab. Per-Ove Törnqvist, who is a research associate, has been working with me for many, many years. Laura Vossen, Ariana Kokko, they have left. They got their PhD some years ago. This was a female student who did a master in my lab. This is Natalia from Poland, who is doing a master in my lab at the moment. So we are usually something between 5-10 people in the lab. And of course, we need money, and we get money mainly from the Swedish Research Council, also from the European Union sometimes. Okay, so thank you very much for listening, and I am ready to take questions if you have any.
So thank you, sir. Thank you very much. I am truly humbled by your numerous insightful contribution to this lecture. And a contribution of such caliber can only come from a true genius like you. Thank you so much for teaming up with us. So at this moment, I'm not getting any questions in the chat box, but actually I have some questions. Actually, so regarding that, it's a very good model system for the preclinical studies of the zebrafish model system. So, sir, you can tell me, sir, what is the lifespan of the zebrafish actually?
The lifespan of, I mean, we can keep zebrafish sometimes in the lab, which are 3-4 years old, but in the wild, I think they have a shorter lifespan. I guess 2 years, 3 years, and we don't want to use fish when they get too old. So the fish we use in experiments are usually between, if we work on adult fish, it's between half a year and 1.5 years or something.
Sir, another question one that it's a very good model for the anxiolytic drugs as a model system. Actually. So if any drug is going to be synthesized and it's claimed for an anxiolytic drug, so how the particular drugs are going to be introduced in the zebrafish actually?
What we do usually is to put it in, the easiest thing is if it's water soluble, so you can put it in the water and you actually have the fish to swim in the substance to take it up.
Yes.
That's not always possible, of course, because not all substances are water soluble, and then it becomes more of a problem. You can put it in the feed, and you can also inject. I mean, we can get you can do an IP injection in the fish. So there are other ways to administrate, but the easiest thing, especially if you work on larvae, which a lot of people do when they screen different compounds, then you have to put it in the water. You can't do really injections in larvae. That's quite tricky.
It's a very good model system, sir, that I came to know. Actually, previously, I have a very little bit idea regarding this, but today it's a very elaborately you are going to discuss regarding these things, regarding the serotonergic and the cholinergic systems are to the civil. Thank you so much, sir, for your insightful lectures.
Thank you.
Thank you, sir. Thank you, sir. So now I'm going to leave, and sir, thank you again for your participation and teaming up with us. Thank you. Thank you so much.
Thank you. Have a nice conference.
Thank you. Thank you.
Good afternoon, everyone. Thank you, Professor Winberg and Dr. Roy, for giving us an enriching experience. Now, let us take a short break and resume back within a span of half an hour. I hope all of you have enjoyed the enriching as well as splendid plenary lecture session. Thank you. Please make sure that all of you join back for the second plenary lecture session of the day within a time span of half an hour. Thank you so much.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as an area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar, and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am an NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm.
বুঝতে পারছি না ঢুকতে পারলাম না কানেক্টিং গড়বড়
আপনার ল্যাবটা তো কারণে কেউ ডিস্টার্ব করছে না এখানে ঢুকতে নেই না আমাদের তোরা কি গুগল দিয়ে ঢুকলে হ্যাঁ না জিমেল আর চেক কিছু করতে না খালি ক্লিক করতে হবে ক্লিক করলাম তোরা দিয়ে তো 2 দিলাম
from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am an NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar, and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now, I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello. Hello.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. So right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and
Hello. Welcome back to the second half of the conference. I, Dr. Moinanman, take the pleasure of welcoming Professor Dr. G.D. Gupta, Director cum Principal, ISF College of Pharmacy, Moga, India. His session will be moderated by Dr. Shutova Bishwas Maji, Associate Professor, NSH School of Health Sciences. Dr. Maji has received her doctoral degree from Jadavpur University and has carried out a postdoctoral research on parasitology, phytochemistry, and microbiology from Bose Institute and Jadavpur University. She has a teaching career of about 16 years and has published around 75 peer-reviewed research articles and reviews in various national and international journals of repute. She has also authored and edited more than 5 book chapters which have been published by renowned publishers. Her research interest includes the exploration of various natural and synthetic polymers in modulating drug release from various dosage forms. She has a specific interest in the development of novel formulations including osmotic tablets, liquid solid compacts, and microsponges. Now I would like to request Dr. Shutova Bishwas Maji to kindly introduce Dr. G.D. Gupta for the upcoming session.
Good afternoon, everybody. Thank you, Dr. Mal, for giving a nice introduction. I welcome Professor G.T. Gupta to the session, and let me introduce Dr. G.T. Gupta. Professor G.T. Gupta is currently working as Director and Principal, ISF College of Pharmacy. He did his pharmaceutical education, both B.Pharm and M.Pharm, from H.S. Gaur Vishwavidyalaya, Sagar MP, and PhD from Department of Pharmaceutical Sciences, Devi Ahalya Vishwavidyalaya, Indore. He has contributed more than 2 decades of dedicated service towards pharmaceutical education and research. Professor Gupta is a prolific writer of professional books and authored 19 books, published research papers in various reported peer-reviewed journals. He has also been instrumental in getting 6 design patents and 3 filed patents, 3 transfer technology, and received research project grants from many professional bodies such as AICT, BST, DPT, CSR as principal investigator, OPI, and coordinator. His contributions have fetched him several national and international awards. And besides being in academics and research, Professor Gupta is a motivational speaker and a social worker. Professor Gupta, please start your session.
So good afternoon, everyone. So first of all, thank you very much, madam, for a nice introduction. And I am thankful to Professor Dr. Subhas Mathiji, Director, NSHM Knowledge Campus, Kolkata. And I am also thankful to the all organizers and management for inviting me and provide the opportunity to interact to all of you on the online mode. So madam, as you have already mentioned that I am giving the motivational talk. So this lecture is divided in the 2 parts. One is the small scientific and actual base what we are the doing, because I know that the topic selected by the organizing committee, this is the very important and very nice research advancement, resilience in the pandemic era, a drive for the innovation and transformation. So we know and everybody know that in those days everybody is the working on number of the parameter during the pandemic, and I have come to know what is our preparation being a research. I know that thousands of the institutes, they are working at a different level, but during the pandemic situation we come to know that only 2 and 3 companies, they are the working in the PPE kit. Only a few companies, they are the producing of the mask like that thing. So nowadays we are independent and we are doing the work in such a manner so that we can produce the thing which are required to our country and to our society. So the topic selected by me, I am audible and slide is visible, madam.
Yeah. Perfectly, okay.
So research advancement resilience in the pandemic era. So what is the research and what is the utility of the research? And if you can think about the post-graduation program and research, so most of the person, they are involved to complete their research project during the 1 year or the 2 year and whatever is the required. But what is the utility and what is the importance of the research project to the society and how you can contribute to the society or the institution and contribution in the research? This is very, very important. So research is defined as the creation of the new knowledge or use of existing knowledge in a new and creative way so as to be generate new concept, methodology and understanding. Let us consider you can think over about your institute or any institute. If we can move through the any one lab, so what every year we are included in a part of research. Either it may be concept based, methodology, understanding, equipment, design, and any way how we can contribute the system, how we can contribute in the research. So this could be include the synthesis and analysis of the previous research to the extent that the idea lead to be new and creative outcomes. So this is very, very important. And these are the very simple lines given by the everywhere. Self-reliance, Atmanirbharta, and need is the mother of the invention. No doubt. And this definition has come to very, very powerful during the pandemic situation. And we are unable to control even today. We have no any treatment guideline of the COVID-19. Number of Ayurveda companies, they are the working at a different parameters. One of the product of the NOQ19, and he launched in the market and mentioned that this is the use for the treatment of the COVID-19. It can be easily, immediately it can be mentioned that this is not for treatment and this is for the prophylactics, or you can say preventive, or you can say protective, because even today number of the researchers working in the different laboratory but no any specific dosage form, tablet, capsule, injection or anything we cannot write down for the COVID-19 because the research parameters are incomplete and testing is the incomplete, and sometimes we are unable to reach in a particular point. And NEP, this is the National Education Policy 2020. The basic concept is the outcome-based education and research. Outcome-based education and research. So everywhere and all parameters are the moving around of this one. Either we are doing the small related to the agriculture, related to the food, related to the pharma, related to the cosmetics, and related to the any area where we are the working. What we are thinking about now, what is the outcome? That is why number of the agency they are the working on. Creativity through flexibility. This is the resilience, the creativity through the flexibility. So nowadays, it is essential and important. What is the need of your research? Why are you performing? So these types of the question and these types of the concept, they are required to be developed in your mind. It is the economic mode because government of India is doing only the two parts of the work. How you can economize. Second one is the what is the outcome, and your outcome is suitable for the society or contribute something or not. What is the consistency, reliability, reproducibility? It is a time-bound program or not, easy to handle or not, disposable system. In those days, itself a very, very big area for the research. And now, number of the newspaper, number of the agency, they are mentioned over there. There is no specific guideline for the disposal of the PPE kit, mask, gloves, even that. So nowadays, surrounding of the hospital, there is a lot of problem. So it is also one of the criteria of the research, and automation is the one of the very important part. And our worthy Prime Minister Narendra Modi ji always mentioned that local to vocal and local to global. So all the research is the required to utilize the surrounding areas difficulty include and work on it. That is why Punjab government, Punjab State Council for Science and Technology, recently they launched one of the idea, and they have given 80 lakhs rupees for the skill development in the surrounding, because we are producing qualified person. What they are need the skilled and research oriented person, they are very, very less. If we can compare with the China, China is the, there is the 98% of the person, they are the skilled. What in India, this is below to the 5%. So now need lot of work in the skill and utilization of your particular area. So not only this, we can work on it, utilize in the for the local media, and then we can also globally we can give the information what we are the producing. Background of what the getting the idea. So if you think over it, first these types of the question came in the mind, how you can, who it for what purpose you are doing, what will it do. Let us consider either you are working on a synthetic part, either you are working in the dosage form design, either working of the any of the testing like of the thing. So what will it be do? Already system is the available, which are the potential market, any unique features of your research, either it may be the polymer, it may be the technology, it may be the stability. Now number of the scientists, they are the working on the nanotechnology, pharmacosome, niazome, and so many things. And in those days, the recently number of the people, they are the working on also on the artificial intelligence. And this is the most probably very, very popular in the engineering side. So what's the unique idea of your concept? Is there enough demand and need of this one? And can customer affordable or not affordable? Why will they can buy it? So these types of the question before to develop any types of the product or any research, these types of the come about or surrounding to you. What is your motivation for doing it? What are the thinking about the product and services? How simple or complex will be the idea execution or implementation be? What are the risk factors involved in the executing the idea? How soon could the idea be put into the operation? What is the break-even point and estimated time frame? So these questions I have not taken from my own. This is very, very important. Gentlemen, these questions I have taken from the MSME national body. And nowadays, micro, small, medium enterprises, they are providing 15 lakhs to the 2 crore rupees for the various ideas. What they need to this one, first you can justify. So I come with one of the example with the MSME during my lecture. So after all, what do you want? After all, each researcher, each academician, they want some of the job role, some of the fund from the various agencies. So how you can obtain the fund? So the fund is required. These types of the question, they came in your mind and you can justify properly. Then I know that your idea, it will be the successful. So government of India, he launched number of the protocols, number of the agency, either you can say AICT, SERV, DST, DBT, ICMR, number, all agency, they are the providing the grants for the research, especially for the COVID-19, either related to the COVID-19 or it is related to nearby of this protection to the COVID-19. Not only that this purpose, they are also producing if you want to design some of the instrument. So if you can design the instrument, it can be helpful to the people, because some of the people, they are the staying in the places, it is very difficult to give a proper treatment. So these questions in the first page given in the Business Incubation Center, micro, small, medium enterprises for submission of your idea. So need to be the clarify and number of the other one. So these are the characteristic of the research, how you can correct. So systemic approach must be followed for accurate data. During the invitation, I have discussed with the Subhas Maitri ji, that's key in those days, What is the attitude of the students? The attitude of the student, they want to the work as a fast mode and very without knowing the historical development, without proper reviewing of the research, they want to do the work. And as you know that initially, if you can go back to the 5 or 7 or 8 year, one guideline given by the UGC of the API, all the scientists move forward toward the API, how we can increase our academic performance. Nowadays, nobody is talking about the API because nowadays people is the move toward the Scopus, web sciences, impact factor, and so many things I will try to discuss over here. Researchers need to practice ethics and code of conduct. This is very important, and this is the responsibility of the individual guide to work on it and maintain the ethical values in the research. Research is based on the logical reasoning and involve the concept. So basic science is the very important, and we need to the include the data or knowledge that is derived in the real time form of the actual observation, depth analysis of the L data collected, and there is the no anomaly associated with it. Nowadays, number of the pharmaceutical companies, herbal companies, they want to testing of the toxicity study. I have obtained of the one product, one of the very big company of the Ayurveda, and they want to the toxicity and they want to publish the paper in a good journal because they know that without data and without justification, we can't say that what amount it will be reached in the blood circulation for the therapeutic level. It is the useful or not useful like that. So toxicity is the very important component of any research and need to do. Existing data helps create more the opportunity for the research. Research is a analytical in nature, and there is the no ambiguity in the interference is the required. Accuracy is one of the most important aspect of the research. So these are the review of the literature. I don't want to discuss now some of the very important point and need to justify before to start of any research work. After discovering, after defining in the research problem, research should make a formal statement of the problem leading to the research objective. During the number of the meeting with the Professor Y.K. Gupta ji, he is the dean and he was the former dean of the AIMS Bhopal, and now he is the member or chairman of the number of the bodies. Number of time I have discussed during the meeting, and number of time he is the visited this institute. He has mentioned that number of the researcher, they are something, they are proposal, but there is the no rational, there is the no justification of the concept. Without rational, how we can launch or how we can give the any sanction amount to the researcher. Nowadays, one thing they mention over that, what is your previous job work, previous work of particular research, because without background of the previous research, it is very difficult to justify. So what is the missing or what are the open question of the study? This is important. Identify the gap in the literature and emphasize why it is important to address these gaps. So this will be form of the rationale of your study. The rationale should be followed by the hypothesis and objective. So three things is the very important. One is known as the rationale. Another, what is your hypothesis? Another one is the objective. On the basis of all these three, rational, hypothesis, and objective, one question is generated. This question is known as the research question, and you will perform your activity, you will perform your work on the basis of the justification related to your research question. What is the research question? What do you want to perform during your research work? It may be the 1 year program, 2 year or 3 year program. So what does justification need? What is the rational? Rational, it will be always very important. And objective, I have given also in the Hindi, the uddeshy kya hai, kyun karna chahte ho, will precisely say what should we research to delineate the type of information that should be collected and provide a framework for the scope of the study. So the best expression of a research objective is a well-formulated, testable research hypothesis. It means what hypothesis you can think it can be pushed to the objective of the research. And a hypothesis in a unproven statement or proposition that can be refuted or the supported by the empirical data what you have obtained and you have studied during the review of the literature. So review of the literature is required very systematic manner study. So hypothetical statement are such a possible answer to a research question. I have already mentioned that one after these three things, we can come to know that what is the research question and what is the research question is the important. So this is the research question answer is very important. We need to give the answer research question. Either it may be the hypothesis, objective, and what is the rational. So these three things what we can the confirm, then we come out one of the things which is known as the research question, so that you can start your journey with your research question with the question mark, and after the completion of the work, it will be the partially full stop. I never say that any research complete during the 1 or 2 or 3 hours or 10 years, because this is a continuous process for the improvement, for the development, and for the advancement. That is why research question start with the question mark, and partially it will be a complete why your efforts, why your dedication, and why your work in a certain limit at a particular point. Identify the problem. So the first and foremost task in the entire process of the scientific research is to identify a research problem. A well-defined problem will be lead the researcher to accomplish all important phases of the research process, starting from the setting objective to the selection of the research methodology. Researcher need to identify non-research problem and research problem. This is very important. Of course, I just want to give one of the example. Nowadays, thousands of the proposal, the peoples they are working during the postgraduate program, they have no any value, and their result already they know very well, even that they are the performing the research. So let us consider here some of the example. Outbreak of the cholera, for example, here that require any research to arrive at solution. So no research means why we want to waste our time. Outbreak cholera, for example, following a severe flood is commonly phenomena in many community. The reason for this is known, and we are the working on. Sometime in the we can say that nowadays some of the problem generated by the floods, or sometime it can be generated by the another region, and we have started the research on this part. This is not suitable for the research question. Found that 1,000 women were continuously users of the contraceptive pills. What last month's services statistics indicate that none of these women were using the contraceptive pills. Well, the fact is the monsoon flood was prevented all where supply of the pills like that. Let us consider if the any drug product, or it can be the lot of the market, but it can be suddenly start. We have started the research, some of the specific problem maybe take place. But the problem is this one, we are unable to supply the product from one place to the another place due to this reason. What we start, we have to the research like that. A hospital treated a large number of cholera cases with the penicillin. What the treatment with the penicillin was not found to be effective. So do we need research to know the reason? There is the one single reason that Vibrio cholera is not sensitive to penicillin, and therefore this is not the drug of choice for this disease. This is also known as the research problem. Let us consider if we can take in the mind, if we can give any penicillin, it will be the suitable for treatment of particular types of the infection. What it means, not that in those days, even you have come to know number of medicine, remdesivir, it may be the use in the during the COVID-19, and it can be available in the market in the lakhs and a lot of you can say black market of the remdesivir. What happened that no any doctor, no any professional body, and no any agency given the name or the recommended of this drug, but we are the using of this one, and the lot of the black money, blacking on the drug product maybe take place. and number of time we feel that one of the news cutting is there. One person, they have produced the, you can say adulterated, or you can say only simple of the dosage form, and fill in the remdesivir container, put on the label and launch in the market. This is the actual news of the paper. The person who has taken placebo like of the remdesivir, they are the protected from the COVID-19, while this person is the in the prison. So this types of the news and this types of the things, it may take place during this period, and number of the professionals, they have developed or they have the published thousands of the paper, even they never work on the COVID-19 like of the disease, never work on the virus, never work on the microbial contamination like that. So these types of the problem may be take place if we not select the problem of the research in a justified manner without using what is the hypothesis, rational, objective, and after that we can come out the with the research question and what is the utility. So this types of the things it may be happen, and during the pandemic situation it will be the increase at a very high level. So research problem is a primary concern to the researcher. A research problem is the perceived difficulty, a feeling of the discomfort or the discrepancy between the common belief and reality. It may be possible, sometimes you can think over it. It is the useful for particular types of the disease. On the basis of this one, you will take the decision, and after the taking the decision, you can follow it accordingly. So sometimes your imagination, your hypothesis, it can be work or not work. What you can think over in a particular and systematic in a line, then it will be the helpful. So problem will be qualified as a potential research problem. This implies that there should be a difference between what exists and ideal or planned situation. What is the available and what are you planning and what is the ideal situation for this one? This is very important. Why the discrepancy exists? This implies that reason or for this discrepancy in unclear to the researcher. One of the industry people just met to me, Vice President of the IPCA and also Dr. Khoplezi of the Vice President Sun Pharma. Both we are on a panel and we are discussing like that. Number of problem in the even in the industry. So people are taking the big problem while they are required to sort out the problems which are very important, like some of the doses form very high and it is required to be reduce the doses. Let us cut the 1000 mg. So if it can be reduced the 500, 600 like that, it will be the applicable. So how you can reduce this one? We need to work on. Even that we have the lot of technology, lot of development, even that conventional drug delivery system, novel drug delivery system, even that very popular, because we need the economization as well as stability and reliability and acceptability of our peoples, those who are staying in this country, and we need to develop accordingly. So the theoretical and practical consideration also play a vital role in choosing a research problem, societal need, and guide to choose a research problem. So these types of the parameter we need to discuss before to start this one. So outcome base of the research is the very important. Most probably we can divide the whole of the situation to 3 parts. Initially we can develop research project and we can move the publication for the national and international. This thing we can put in the mind how many paper we can publish. Research even not we have start, what we have the fix our idea how many paper we have published after the completion or during the research. That's okay, you can think over it, but without any data available with you, how you can justify? Now we are the more over focusing on the scopus, impact factor, high impact factor. We are the need wave of science, and commonly applicable we are the using the I10 index, H index, G index. This types of the thing is the discussing and question asked during the if you can appear any interview. So what is your citation? How many paper you have published in the impact factor? So, gentlemen, I am giving one of the presentation in AI City. So AI City panel person asked, Dr. Gupta, your institute published lot of paper, everything, that's okay. You just give me one of the things. And this one thing is the how many monograph you have published in IPBP or US. Because if you are the researcher, if you are the doing lot of work, you are publishing the paper of the more than 100 impact factor in a year, 200 impact factor, then your research is obviously to contribute to the society, to the country level or in the international level, means need monographs. So gentlemen, now the things is the going forward very, very fast. The away from the your thinking like the only the impact factor or like a scopus. And another question asked to another question to me, and this question was the what is the commercial value of your patent? I have come to the number of the university, they have mentioned that 450 patent, 300 patent, 200 patent. What is the value of these patent? The value of the patent, if you have only the one patent and it have some of the commercial value, then this is very important. What is your consultancy? How many times you can visit the industry? How many industrial problem you have handled? So it is not easy job, I know very well. But if you are work on the research and need in this pandemic situation, we need to work out on so many parameters. And during this pandemic situation, government of India and various agencies, they are giving a lot of funding for the research work. This is up to you, how you can work and obtain and utilize. So monograph, patent, transfer technology, consultancy and commercialization. So in those days, our professional bodies and we are the working on it, how much we are using the consultancy part for your technology. So how much transfer of the technology during the 3-4 year, why your institution, and this is the viable and not viable. What is the consultancy near about the research project or the problem, or you can say thrust areas where you can work or which can be given the problem by the industry, and the commercialization techniques or commercialize, you have commercialized any product. This is very, very important. So nowadays, though professional body which are supporting institutional innovation cell given by the IIC for the entrepreneurship startup, we will all are doing the work. Business incubation MSME, today I will try to discuss this over. Life science sector, this is LSSDC, and entrepreneur and many professional body I have given this one. So MSME in Punjab only total I think is the 7 business incubation center. One of them is the ISF College of Pharmacy. Total incubation center and throughout the India, not pharmacy, including overall, near about the 400 plus. And let us consider if your institute have the business incubation of the MSME approved or not. What if the nearby, if your institution or university, they have the MSME business incubation approved, then you can submit the idea. Every 3 years, every 3 months, they invite 10 ideas. And one idea they have provided the 25 lakhs rupees, 15 lakhs to 25 lakhs rupees, 15 lakhs for the state level MSME, and the central level MSME project, they can be provide the near about the 20 to 25 lakhs rupees. So these rupees is not for the to restart your journey. What is your idea? You can execute in the value of the commercialize. That is why at the initial slide, I have taken near about 25 questions. And all these question answer is required to give in a justified manner, because government providing fund to you to generate, to develop one of the product, one of the idea. It is not essential that you can do a big work. I was the present in the one of the meeting of the MSME. One person is develop only the small product, and this product is used in the this is the machine, so that thread it cannot be the break easily. And they have given idea, and most probably the Singer or Usha company, they can be purchased the idea, and they provide the lot of the grant like that. Nowadays, this types of the things important. So 10 idea at a time you can be implemented. One of the business incubation center every 3 months. So every 3 months means near about 40 ideas you can include in a year. And not only this, if your business incubation center 2 idea approved, government will provide to you 1 crore rupees for development of the excellency of business incubation center at your institute. And they have required not near about the 2,000 square feet of the constructive area which is required for the establishment of the MSN. Zero effect and zero defect, this is the jet. This is the now everywhere is the applicable. While this is the only applicable in the industry, but why not in the education? Why not in the research? Why not in the everywhere? Zero defect means this is the jet, and this slogan given by Prime Minister Narendra Modi ji. Zero defect means quality of product has to be very high and free from defects. Again, I repeat it, the product must be free from any defect, and it will be the durable, it will be the effective, it will be the useful to the society. Zero effect means that there should be no adverse effect on the environment. Why the manufacturing? In those days, what we are the doing? We are the doing, and we are the highly selfish. What is the utility and what is the need of our institution? What is the need of our house? What is the need of our industry? And we are not aware about what we are the byproduct left over it. Let us consider we need to protect our environment, because number of the people they are staying there, including yourself is the also. So either water, either air, either electrical, either any types of the gadget, or you can say electronics material, it cannot be harm to the environment. So JED is the one of the most important powerful tool if you can use even the education, means zero defect and zero effect. Means number nowadays I also include one of the things, we work on our student without any physical and biological harm, and we need to give a correct situation and correct value so that they will be grow in a very fast manner. Here this is the I have what I have obtained, and I want to give the example is the here this instrument design in our institute, and I have obtained 18.5 lakh grant from the MSME. That is why I want to put and pushing to all of you, you need to submit your idea. 10 idea at a time, one institute can be submit, and our 2 ideas selected. By the way, second idea is the under process, and I have obtained the this amount 18.5 lakhs from the MSME to develop this product. And we in the pharmacology lab we use the Morris based maze, analgesiometer, elevated plus maze, grape strength. These are the 10 instruments we are using separately. What? Our faculty member, Dr. Siddharth Mehan, and his research team developed this instrument. And I hope that very soon it will become in the market. And I will try my best to launch this project in 2020. And most probably our team is the work on it. And this is the only the model 3D prepared by the computerized, and he is also prepared why using of the raw material put in like that. Now it can be converted in the form of the fibers, and what things we can use as a automation and digital, we will try to include these types of the things in this. So this is the specification, and why I am using this one. These types of the idea I have submitted, and nothing, that's all, only 3 slides. And these three slides is the specific because the utility of the instrument. It means not that you can develop only the dosage form. There is the number of good things. How you can justify utility of this one, either in the laboratory, either in the regular practice, either in the services or anyway. So it have the 5 arm. This is the 1, and this is the another 2. This one is the 3. and this one is the 4, and this one is the 5. So specification and 1-to-1 point is the given over here. These are the chamber A, B, C, D, and E, and designed to elevate locomotion activity and grip activity only in this area. So these are the 5 arm, and these 5 arm suitable to identify, detect, or analyze, or you can utilize to detect all utilization of the 10. It means this the name of the instrument is the 10 and 1, and this is the shape of the ace. This is means as maze, and I hope that it will be coming out in the market after the number of the practical. First we perform the practical on the individually, and the same annual we can use in this one and find out the result and the probability, how many parameters or how many data interpretation it may be take place, and what will be the change it will be happen. It will be, I will try. So these are the scheme of the various MSME. I don't want to discuss now the pharma 4 0.4. So gentlemen, industry is growing very fast, and pharma 4.0 launch our industry. On the basis of this one, pharma 4.0, I have mentioned this of the table. Pharma 4.0. So what happened about the academia? What we are doing the academia? Either this is the at par or equivalent and not at par. So what is the going on the academia? Research and innovation. Let us consider pharma 1.0 means starting point of modern pharmaceutical industry. At that time, most probably we can say manual work is the going on. Tablet machine, capsule filling machine, most probably 90% of the things it will be handled the manually. So this is the 5-5-5-year segment, and recently 5.0 pharma, it can be available or very soon it can be come out. And 2.0 pharma means development of the machine and modernization, and pharma 3.0 means fully automation and continuous manufacturing technology. So multi-rotatory machine, or you can say slightly untouched or partially touched only by the human being, is the fully automation is the 3.0. But nowadays, environment considering this one is the integration of the connectivity, artificial intelligence, robotics, and elevator system that operate with the little no human being. Untouched product it will be available in the market. Any product, it can be from raw material one time use, the finished product with the fill in the container, labeling, and everything, it can be finished form, it can be available. So this is known as the pharma 4.0. And Mr. Rajput in one of the panel, he is from the Kerala, and I simply discussed with them. He told that Dr. Gupta, pharmaceutical education at that time only equivalent to pharma 2.0. And if you want to match this one, so all the academician, researcher, they need to the work and increase the more facilities in the academia, so that we can find out and we can achieve our goal. So academia is the manual process of the teaching learning process with the overhead project. This is the initially, this is the academia 1.0. Academia 2.0, before to the pandemic, we are the working on it. Number of the institute, they have the multimedia facility. Number of the institute, they have the excellent digitalized equipment. After the Pharmacy Council of India, they have mentioned remove the 3-tablet hand operated machine, need to the one of the automation. So some of the institute, they have modified, they have changed, but even that number of the institute, they have the same types of the material. Now immediately some of the lockdown, it may take place, we can shift it from the offline mode to the online. So at that time, partially we prepared our online mode. I want to ask over here, how many institute they have developed their own recording theater, own recording room, so that these types of the recorded lecture we will provide to our student. So this is very, very important. Even that we are unable to the develop. So right now we are at the position of the 2.5 of the academy, and research and innovation, we are reach up to the 3.0, and 4 point need at that time technology transfer, commercialization, automation, handling of the software. Only few institute they have the some of the institutes, there's a Gastroplex, CAD, QVD. It may be used in the pharmaceutical chemistry, pharmacology, pharmacognosy. Number of the software and technology is the going on, because if we want to perform the research, we are the sending the sampling from the long places, and it will take time of the weeks or some of the fortnight. So even that condition, part of the sample, it can be rapture. I have received one of the sample from the Indore, and they want to know that what is the globule size in this one. And from my side, it will be determined 631 micron. And they feel that it will be the, at that time they have manufactured, at that time this is near of the 10 micron, because as per the FDA guideline, they want to the value of this one, and they feel that the given the FDA guideline, 1 ml contain not more than 5,000 globules, and the size is less than of the 5 micron, because they are the working on the some of the laboratory. Show that research and innovation is near about 3.0. But if you want to the research and innovation 4.0, we need the increased lot of facilities in-house, so that we can develop our lab, we can train our student, and we can provide the proper placement level to our student. So my expectation is the here, and what I feel over here, that the person need to be focused on one area. Either you are developing the dosage form, polymers. So if you can focus only the one area, then you can grow very fast, because in those days, all types of the thing is the required for the development of the pharmaceutical education as well as research. So these point I do not want, and ethics I do not want. So this is the, I have mentioned over some of the few slides. We need to a smart work, because, and we believe in the teamwork. Luka छिपी का काम नहीं चलेगा. So forget it. The time is the gone of the one of the story of the tortoise and the rabbit. And even this story, it reached in the 4 point, and 2 times of the tortoise won the race, and only the 1 times of the race won by the rabbit, and 4th time they have decided that we need to work in a team, whatever you have the strength, because each and everybody have some of the strength and some of the weakness. So we work on our strength and our weakness, so we can come together. So multifaceted development and multidisciplinary activity is the required. So we need to the smart work. We are the hard work is the gone. We are doing the hard work, lot of hard work. This is not possible. You can grow. You need the smart work. And this is the intentionally I have taken one of the photo and one of this one. And this is the एनक जिसको अपन कहते हैं goggles like that. Need to work on your brain. हमें अपनी RAM को ठीक रखना है. Memory, RAM. Lot of stress, lot of depression, lot of anxiety in those days in the students and even that of the faculty. So we need to work on, if we are free from the stress, anxiety and depression, then we can do the work and lot of things. So need to clarify and justify your brain activity. खुद पर काम करने की जरूरत है. Need to work on. And here there is the no any rocket science. Everything it can be passed through from this place. It can be convert this and then this. And there is the no rocket science. If you can thorough, then it will be it. I have mentioned is here is the one word. Practice makes a man perfect. It is the one of the old line. Now need perfect practice makes a man perfect. Now this is the overcome. And another technical perfect practice makes a man perfect. So not only this sentence is the very old. Practice makes a man perfect. That's okay. What it is not, you can grow with this one in a long way. So need a perfect practice makes a man perfect. Not only this, we need the 3 things: technical, perfect, and practice makes a man perfect. Not only this now the need is the specialized. We are working on the specialized training, specialized, technical, perfect. Practice makes a man perfect. Check yourself where you are. You are doing only the practicing. You are doing the perfect practicing. You are doing the technical perfect practicing, or you are doing the specialized training, technical. Nowadays, recently, one of the meeting organized by Pharmacy Council of India, and they are working on the super specialization. Not only the M-Farm, above to the M-Farm, they are working on the super specialization, and one team and one of the committee is the constituted to work on it, how it can be useful and fruitful to the industry people for the super specialized. A good teacher or researcher is a master of simplification. Again, I repeat it, a good teacher or researcher is a master of simplification of science and technology in the benefit of society. A good teacher or researcher is a master of simplification of sciences and technology in the benefit of society. Then you will be the successful. Here is the don't burn your opportunity for temporary comfort. You need to don't wait for opportunity. Create your journey and develop yourself. Take the whole responsibility on your old shoulder. Don't you give the responsibility or the blame to the others. You need to work on your research work or technical. So this lecture dedicated to my guide, Guru Professor R.S. God. Teacher need to change mindset, perception and encourage student. Again, I repeat, teacher need to change mindset, perception and encourage student to grow and grow. So I hope that you will be the take or enjoy this lecture, and I hope that this will be the helpful to you in the research point of view, motivational point of view. When you are the working in the laboratory, it will be helpful to you. So thank you very much.
Thank you, Professor Gupta, for your nice deliberation. Actually, your lecture, I found it very useful, not only for the student community, but also for the students who are going to join research very soon, young researchers, as well as young academicians who are there in the field of research actively, and also those who are interested to join as entrepreneur. So because nowadays in the COVID-19 era, and also our honorable Prime Minister has also is stressing more on Atmanirbharata. So your lecture is just in time, actually giving a lot of enthusiasm and inspiration to all of us. So you have really focused on various things like that before starting any research work, a researcher should be able to answer to all the 25 questions you have put down. Actually, when any researcher can frame the questions and answer the questions, all the questions, then only he can justify his research work and can be able to procure any funds or grants from any professional body like you have mentioned. And this MSME actually is a very new concept. I think which will be a very helpful to all the researchers who are interested to have a venture of their own. And
হেডফোন দিয়েও আমি কিছু শুনতে পাচ্ছি না শুধু ভয় পালা কি হবে
and also you have stressed on the outcomes of the research in terms of not only research publications but also patents, and that patents also of highly commercial value. Now that is very important. Many of the researchers, they are more focused on publishing their research work in peer-reviewed journals and journals of high impact factors, but unless they are of any benefit to the society, at the end it is of no use. That point is very clear to all of us. And you have also mentioned about this scope that nowadays if any academician wants to get a good job, he is not asked about other things but about how many citations, Scopus index, then H index, I-10 index, etc. So I think this your lecture has been very helpful to everybody. And I haven't found any question in the chat box yet. So, Professor Gupta, if you want to add any further point on your lecture, please.
So, madam, just I want to feel that during the our journey of the 25 year of experience, what I feel that most of the person they want to the shortcut, and due to the shortcut, the fruitfulness of the research is not justified. Second thing, midway journey, we have start our journey and we left our journey at the midway. So due to this reason, the wastage, lot of wastage of the money, either it may be the institute or it may be the government, or you can say from your own pocket. Let us consider any person, if they have start their work. Nowadays, our student, they have the working on some of the product, and the product applicability is this one key. Let us consider nowadays, you know that a lot of problem related to the farmers. So farmers, why they are the doing this types of the things? So we, our people is the also working on this part. Let us consider in Punjab, we are producing in the bulk amount of the rice, and due to the production of the bulk amount of the rice, what happened that the cost of the product, it is obviously reduced. First thing. Second thing, government, how much amount of the work government they can be stored at their warehouse. So that is why in those days, government of India is also focused, we can distribute the things. Let us consider, if you have the 10 acre of the land, you can divide accordingly so that fertility it will be the maintained, as well as the crop cost it will be the increase. So happiness obviously it will be the increase. That is why our peoples, they are the working on the testing of the grains and the how we can add the value on this case. And another thing is the herbal point of view, if we can think over it. Nowadays, lot of problem in the herbal and on the pump and so like that. If you can show that, एक वो कहा जाता है कि बाबा होते हैं, एक शिष्य आता है, पैर छूता है, बाबा आपकी कृपा से ये हो गया, सेकंड थर्ड फोर्थ आते हैं और बाबा ऊपर चढ़ जाता है, एक आश्रम बना लेता है. In the same way, that one, without any testing, how we can develop or how we can grow. That is why nowadays number of the Ayurvedic companies, they are moved toward the testing. They are the move toward of the data analysis basis. Recently, I have visited the National Institute of the Ayurveda, Jaipur. As you know, National Institute of Ayurveda, it is the one of the biggest unit, and they are producing the lot of things. And first time I come to know that how to purify the mercury. I don't know any one person if know the mercury, it is required to 48 hours to keep churning of this the mercury with the 10 different material. After that, it can be useful for the medicine and the purpose of the treatment. So number of time, either honey production, either production of any types of the herbal crop. So this types of the testing is the required in each and every state. Some of the state, they have the water problem. Some of the state, they have the high, or you can say high temperature climate condition. Somewhere is the heavy rainy season. So our people need to the work on it. Not only this, some of the disease, this is known as the seasonal disease. How many person they are the working on the seasonal disease, so that people they can be protect even simple malaria, and we are the not working on. हमारी कंट्री में तो ये साधारण सा काम है कि बरसात खत्म हो गई है, अब ड्रेनेज क्लियर हो रहा है. So I don't know why we are not to the prepared and the before to this one. That is why this types of the problem is the going on. So from my side, and we are the ISF College of Pharmacy, we are running near about of the 6 crore project. And these project, basically some of the herbal products, we are the testing is the going on, animal testing is the going on, some of the skill related is the going on, and some of the product we are launching. One product we have the given transfer technology to the fill gap of the India. This is the mouth dissolving, this is the film, and one of the another product. So our student, even then M-Farm, we are the more focused on the industry peoples. And most probably nowadays, most probably I am direct interacting to the industry peoples. Even these peoples don't know what is going on the institution or the academia. I simply put one question to the one of the pharmaceutical person कि जो हम कर रहे हैं वो आप मानते नहीं हो, और जो आप चाहते हो बताते नहीं हो, what I will do? So every time you can say that there is a big gap between the industry and academia. So how we can fill this gap back? When I was even in the VFAM in 1993, at that time our teacher, they are the working on workshop, seminar, symposium in the industry academia interaction. At that time they are the doing. And after the 25 year, even that we are working on the same thing. It means somewhere is the big gap. And if there is the no big gap, तो या तो हम कुछ छुपा रहे हैं या वो कुछ छुपा रहे हैं. तो ऐसे काम रिसर्च का नहीं चल सकता है. ये मैं ट्रू फॉर्म में बात कर रहा हूं. Because we need to the clarity. Industry don't want to entrap the academia people, and academia people don't want to entrap with the industry, because we are not matching with these two things. So nowadays time is the gone, time is moving very fast, our industry growing is very fast. If you want to place your student at a good position, we need our train to our student at part the requirement of the industry. And I have mentioned that one word to the academy peoples कि सर वो कौन सा दिन होगा कि मैं अपना होर्डिंग लगा सकूं कि बी फार्म स्टूडेंट ऑफ 10 लाख पैकेज. कब आएगा वो दिन? इंजीनियरिंग के बड़े बड़े लगे होते हैं, क्या हो गया है? 10,000 रुपीज जॉब के लिए हम 4 साल टेक्निकल पढ़ाते हैं. इट मींस समवेयर इज द प्रॉब्लम. तो ही इज द सरप्राइज दैट तब उन्होंने कहा था जस्टिस गुप्ता, now we are the launching of the pharma 5.0. what is about your in academia? You are even not as 2.0. That is why I have mentioned this slide. Gentlemen, if we are the academician, we are training, we are producing the student for the industry, so why not we can do some of the club activity so that our student, they will be the trained at par of the industry, and we can say that our student at par equivalent to the industry peoples, and globally they can be spread out. So lot of things is the available in the market. हमने आपको LSSDC की बात की, MSME की बात की है. MSME is the one of the big hub. I don't know NSHM it's the have the business incubation center or not. You can apply to the for the business incubation center, develop the incubation center in your institute, and every 3 months you can submit of the near about 10 idea. With the budget of the each idea is the 18 lakhs to 25 lakhs, and whenever you have obtained the 2 idea, they will provide to 1 crore rupees for the development of the infrastructure for the business incubation center. Because I know that if we can produce one pharmacist only help to their family, a small family or like a mediocre family. If we can produce one of the entrepreneur, it will help to the 100 of families. So this is the requirement of Indo Day, at least 1% of entrepreneur or 1% of the innovative person we need to produce every year from our institutions.
Thank you, Professor Gupta. Actually, that is the main important thing, that there is a big gap between the industry need and the curriculum of the academic institutions. That has to be merged very quickly for the benefit of the society, for the benefit of the young generation who are highly interested in pursuing the pharmacy courses. So it's nice to have you as a speaker with us. You have really motivated all the audience with your nice speech. Thank you, sir.
Thank you very much, madam. Thank you.
Thank you. Thank you.
Can I leave now?
Yeah, thank you, sir. You may leave. Thank you, sir.
Thank you, Dr. G.D. Gupta, sir, and Dr. Mashi Madam for this insightful session. I'm sure all the participants are enlightened as well as delighted at the same time. We will have a short break now of half an hour and resume sharp at 4 p.m. Thank you all.
Really blessed that I'm in such minds. Right now, I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Medtronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am in such minds. Right now, I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am an NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am an NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. So right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy pharmacy portal. Right now, I am working as an area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar, and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. So right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Soumyadeep Sarkar from 2008-12 batch pharmacy portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar, and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. So right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Hello everyone, I am Somideep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. Right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
Welcome all to the first panel discussion of the day. The topic of today's discussion is drug repurposing and repositioning in perspective of COVID-19 treatment. I would now take the pleasure to introduce the moderator for the panel discussion, Dr. Vijaya Ghosh, Professor, NSHM School of Health Sciences. She has been teaching for more than 30 years, and before joining NSHM, she has taught in prestigious pharmacy colleges of both North and South India. Her research interests involve antivirus, transdermal drug delivery, and optimization. She has published 41 research articles, authored pharmacy books, and co-edited research books for reputed publishers including Elsevier. She is also keenly interested in creative writing and has published her work in both Bengali and English languages. Now I would like to request Vijaya Madam to kindly introduce the panelists and start the session.
Hello, a very good afternoon. COVID-19 appeared when it was least expected. In no time, it turned into a tragedy because of the exponential rise in cases and fatality. In many parts of the world, medical systems broke down. In absence of a treatment option, World community was struggling to manage the situation without a proper drug. In this dire emergency, when little was known about the disease and not even a single drug to fight it, the onus of finding a cure fell on the research community. It had to fight it without a proper medicine, with the existing ones only. They had gone for an innovative option, repurposing of the existing ones. In today's discussion, we have two eminent panelists, Dr. Deviprasad Chattopadhyay and Dr. Rubina Bose, sharing their knowledge about this topic. I request the viewers to follow the discussion, and if you have any question, drop in our Q&A box. First, I will introduce Dr. Rubina Bose. Dr. Rubina Bose is a regulatory person. For the last 2 decades, she is with the National Regulatory Authority of India, Central Drug Standard Control Organization, CDSCO. She has worked in various capacities, but one of the responsibilities she has carried out is quality assurance of vaccines. Presently, she is stationed in New Delhi, looking after international cooperation of CDSC. Dr. Rubina was also a technical officer in whose pre-qualification vaccine assessment team at Geneva. She has traveled widely to represent Government of India in various WHO meetings at Geneva, USA, Thailand, Turkey, and many other places. I request Dr. Bose to take over the session. Am I audible? Yeah. Okay. So, thank you for the kind introduction. And also, it's a thank you for this great opportunity to be in the connection with all the fellow pharmacists, my professional, and other colleagues and friends. So, what I think my introduction was nice, and it was great to have such a kind introduction. Just to say that I'm also a BPharm, MPharm, and doctorate from Jadavpur University. And I would like to understand what was the expectation from this particular topic, which was on drug repurposing and repositioning. So I don't know. I would like to just say some basic things which we have at present, which is continuing at present as a regulatory regime. And you must have seen, as Madam was saying just before this, that we have undergone through an unprecedented pandemic, and it's still on, and unfortunately and sadly the pandemic is still on. And we know that unless everybody is safe, we are not safe. So we keep on saying these things. And as a representative representing CDSU in the various forum, I keep on saying the same thing, and probably working. And today also, like, we have a very important discussions on the COVID vaccines. Proposals are ongoing, and just now we have done with the DNA vaccines. So in between, I just thought that it would be very small time for me to really give a perspective, but I would like to give it in a very simple manner. But before that, I just wanted to say something in background, that long ago, before WHO has declared this pandemic as a this public emergency as a pandemic, it was long before that that government of India has started. So it was declared pandemic in 2000 March 2020, but we started our preparedness. Um, Government of India and along with the regulatory agencies started its preparedness much ahead in January 2020, ensuring that the drugs are available, the ingredients or API are available, and there is no black marketing and hoarding. So there are regulatory advices and suggestions which have gone to um, all the stakeholders, to the manufacturers associations, to all the state governments to ensure that the drugs are available to the public. In this process, there was also the CDSCO, as I work in Central Drug Standard Control Organization. So CDSCO as the as a regulatory authority or as a national regulatory authority has issued number of advices and notices to the various regulators. So because as you know that CDSCO is the national regulatory authority or the central drug control, and we have 35 states with the union territories where we have the state drug controllers. So we all together work for the country as a regulatory agencies. So in this, CDSO has issued advisory notices as well as different notices to all the state drug controllers to ensure the availability of the various drugs, to keep monitoring on this and vigilance on this availability, as well as the public notices have been immediately published as early as in March 2020, 19th March. If you go to the CDSCO website, I don't know how many of you are aware and are used to seeing this CDSCO website, which is the cdsco.gov.in. I suggest all of you to go through that website and see that we have the various public notices published on the same day, that's 19th March 2020, saying how to go about if you have an applications for a clinical trial or approval for a drug for COVID-19 treatment. It may be a drug, it may be a biosimilar, it may be a vaccine, it may be a COVID-19 testing kits. Would like to say that we had not a single kit, COVID testing kits, diagnostic kits, for which was the first thing which was most important for us for diagnosis of the COVID. So that kit, there was not a single kit in the month of March. And in the month of May, we have come up as an India, as you know, we are the hub of everything. Like a pharmacy, we call ourselves as a pharmacy of the world because all our innovators and the manufacturers have come up with the kit, the first indigenous kit in the country. And so there was Smilab manufacturing the first indigenous RT-PCR kits, and there was a line. So at present, probably we have more than 250 diagnostic kits permitted in the country. And when in March, we had not a single one. So, and we also have come through a lot of steps. So in that pursuance, so we had there was a publication of the public notices and the various regulatory guidelines on how to apply or how to go about for a drug and for a clinical trial. So it was said in such publications that if you have a drug which is for the, as I just to say about this drug repurposing and positioning, I just wanted to go to this background and then come to you as a simple definition and what we are doing at present right now, whether we do have such drug repurposing and positioning in place. So, and what is our regulatory enablers. So this is like a simple thing which I wanted to say you. So we say this in this background that any farm or research institute having a protocol for repurposing of existing drugs, vaccines. So we are already saying what does it mean, that what is drug repurposing or what is drug repositioning. For treatment of the COVID-19 will be given a priority for review and approval. So applications for a clinical trial permission, applications to import or manufacture drug vaccine for sale and distribution would be processed on priority through expedited review or accelerated approval process. So what would be the data requirement for such that when we are trying to say that we would be doing everything in an expedited review or accelerated approval process, there was clarity given on how to conduct a clinical trial in this COVID when there is a complete lockdown situation in the country, how to go about with the clinical trial when the where the subjects are, there is a challenge in the different sphere of this, whether the patients would be available, how to go about and get in contact with the patient's population, how to conduct a clinical trial. So all these cases, all such clarification was issued in the form of guidances and public notices. And it was as I pointed out, so regarding even for the drug repurposing, there was a guidance provided. So what is this? So we can just simply say that it is the known drugs when it is being indicated to treat a new indications or a new disease or condition. So the drug which is already being approved for some specific indications, now it is being to be used for another new indications. So we can call these as a repositioning or reprofiling, retasking, or therapeutic switching. So as you know that there has been lot many such kind of drugs, like it has been continued since past, aspirin, which are now being used as an antiplatelet agents. Uh, yeah, sorry, are you writing? Uh, Actually, I have, sorry, I'm not playing my video because I'm in also, in connection with another meeting going on. So if is it okay if I don't play my video? I'll just come back after some time. Right. There are so many drugs like minoxidil, which is being as an antihypertensive, and now it is being used also in the case of the alopecia or the hair fall, or in the thalidomide for morning sickness, and also as an anti-cancer drug. So there are so many, a lot many. There is a queue of lot many drugs which are being used for this such a purpose. So there are questions that do we have this in our regulatory? What is the regulatory pathway for this? Do we have it in our country? Yes. So I would like to say that that's what we have. The regulatory enabler, the new drug and the clinical trial rules 2019, which was published in March 2019. This gives all such pathway where we have an expansion of the definition of the new drug, including so many advanced medical therapeutic products also in this new drug definitions. We had all such mentioning of the time-based, time-bound disposal of applications. Even if a drug is indigenously developed, that means the research and development of the drug is only in India. In that case, if somebody approaches CDSCO, then they can do this, and if CDSCO does not come back to the applicant within 30 days, it is considered to be deemed approval for a clinical trial. So all such kind of provisions are being given in this new drug and the clinical trial rules. Um, and there is also, as I was mentioning, the provisions of accelerated approval, mentioning that in case of considering the severity, rarity, or prevalence of a disease in the Indian population, when there is a lack of alternative treatments or availability or lack of alternative treatments, when it is noted that a particular drug is having a meaningful therapeutic benefit over the existing treatment, in that case any applicant can come to CDSCO and apply for the accelerated approval process, and also can ask for a waiver of a local clinical trial. So how come we go about with a waiver of a local clinical trial? So there are all conditions mentioned in this NDCT rules 2019, where you can say, like I would like to. So if we just a small short, I can say the new NDCT rules we have, like it is in the website. Anybody can go and see this. There are 13 chapters. Though the chapters contains different paras and the rules. We call that these are the rules of the NDCT rules. And then there are 8 schedules in that NDCT rules, in which one of the schedule, which is the second schedule, it says about the process for applications for a new drug for the clinical trials, for marketing authorization,
if you wish to manufacture the drug or if you want to import the drug. So in all cases, what would be the data requirements, what you need, what is your preclinical study, what is the drug development part, what is the data needed for the preclinical or toxicological study, what is needed for going into the next phase, that is the phase 1, phase 2, phase 3, or can we directly go into phase 1, or can we go from without doing a phase 1 or phase 2, can we directly go into phase 3? So all those kind of things are there. So here we mention about the drug repurposing. Am I audible? Sorry. If you want to point out something, you can please, and you can ask me when I need to stop. Is it okay?
Yeah, it's okay.
Okay. So just here, I would like to say this, the second schedule, NDCT rules 2019, has the regulatory provisions to go about this drug repurposing, and or we can also call it drug repositioning. So where it mentions that requirements of the data and information for permission to import or manufacture of a drug already approved, which is now proposed to be clinically tried or marketed with certain new claims, in which what are the requirements? In case of such a drug, so I'm going through the different requirements. In case a drug already approved by Central Licensing Authority, that is CDSCO, for certain claims, which is now proposed to be clinically tried or marketed with a modified or new claims, namely indications. It can be new dosage or dosage form, including sustained release dosage form. You know that modified release dosage form, and the route of administration, or it is a novel drug delivery system. The requirements of the data and the information for permission to import or manufacture of such new drug for sale or to undertake clinical trials shall depend on the nature and the regulatory status of the drug for the new claim in other country. In case the drug is imported, in case the drug is already marketed in the country, it has been already approved by CDSCO, what is there? Application for approval of manufacture or import of such new drug or to undertake clinical trial actually may differ from application for a new drug molecule, in that they allow the applicant as well as the regulatory authority to rely at least in part on the safety or efficacy of the data of the drug formulation already approved, because we already have the drug in place. And if it is the same applicant who is having the same chemistry, manufacturing and formulation data, so there would be a different mode and modalities and a pathway for review of such application. So however, additional non-clinical or clinical data may be necessary to substantiate the new claims, considering some of the following points. What is this? The chemical and pharmaceutical information will be same as prescribed in this schedule. However, the data requirements may be omitted depending on whether the drug formulation is already approved and marketed in the country by the applicant in the same dosage form for certain indication. If it is approved and marketed, no further chemical and pharmaceutical data is required to be submitted by the same applicant. But if it is a different applicant, definitely the data is required. The animal pharmacological and toxicological data and clinical data needed in such cases will usually be determined on case-by-case basis, depending on the type of new claims being made by the applicant, as well as the mechanism of action, pathophysiology of the disease or condition, safety and efficacy profile in the respective conditions or populations, and clinical data already generated with the drug in the approved claim. That means if you have a data, already the drug is marketed, so you must be having a great safety profile of the drug if it is found to be safe. So now what you need to prove is the efficacy of the drug in the new indication. So that is the thing which is being told here. So the requirements may be abbreviated or relaxed or omitted as considered appropriate by the CDSCO, Central Licensing Authority. So if the drug is already approved and marketed in other country for the proposed new claim, in that case, they can directly come to CDSCO and they can ask for a phase 3 trial, or they can ask for a local clinical trial waiver. In such case, how do we consider that whether such waiver can be given? It depends on that if the drug is already approved in such other countries. Like that, we have recently approved the vaccines, like some of the vaccines like the Pfizer and the Moderna. If you have seen that these vaccines have been approved in the country, because this has been already approved in some of the countries like US. So it depends on which countries, stringent regulatory agencies like US FDA or the PMDA or it has been approved in the EMA. In such case, to make available the vaccines in the countries, it has been immediately given permission. And the same thing which is also can be followed for a drug which is under repurposing. In case you have the data available with such agencies, or you need already have the drug permitted in this country, and you can directly go into from the drug submission to the phase 3 clinical trial instead of going to the routine process of preclinical phase 1 and phase 2. So that's the process. So the clinical trial also can have that doesn't have involve a route of administration dose patient population that significantly increases the risk associated with the use of the drug. So these are some of the cases where we can also abbreviate the data. In case of an application for permission to undertake clinical trial of a new drug formulation which is already approved in the country, no chemical and pharmaceutical data and non-clinical and clinical data is required to be submitted, provided the clinical trial is proposed to be conducted with the new drug manufactured or imported by the firm under necessary new drug permission or import registration and license, as the case may be, and it has been granted by CDSCO. So these are some of the cases where we go about. And I would like to just point out some of the drugs which we have been given such permission in this recent COVID-19. As all of you are aware, the favipiravir tablets suspension, which were on the old indication for such drug, this is presently being marketed by Glenmark, and then we have a list of manufacturers who have been given subsequent permissions of these favipiravir tablets based on the under an emergency use authorization. So we call this as a restricted use under emergency conditions. It was earlier indicated for novel influenza strains, influenza that cause more severe diseases, and then now it has been given permission for mild to moderate COVID-19 disease. Similarly, we have the itolizumab, which was earlier permitted for the plaque psoriasis, and now it has been also permitted, this permission being given to Biocon for the treatment of moderate to severe COVID-19 complications. Then there is also the pegylated interferon alpha 2B injection, which was given earlier permission for chronic hepatitis B and C, and now it is being given for the moderate COVID-19 infection in adults. We also have baricitinib tablets for the treatment of moderate to severe active rheumatoid arthritis in the adults, earlier as an old indication, and now the new indication also in combination with the remdesivir is used for the treatment of COVID-19 positive patients in the moderate to severe COVID-19 treatment. So these are some of the examples which I can tell for the current regulatory regime in which we have given this COVID-19 pandemic permissions. And I would like to happy to take some questions, or if you have a discussions, how you want to go about. Thank you so much for giving me this opportunity.
Thank you very much, Dr. Bose. You have actually shared the information that we can only gaze, the long struggle when the country was getting ready to fight the situation, the tiring process of preparing the diagnostic kits, the infrastructure, the recall of the people who can really help. I had some questions, definitely, most of them you have already answered. And one of our panelists, he is actually struggling to join. I talked to him, but somehow, some technical, Devuda has joined just now. So I'll first introduce him and then I'll give the questions, okay? So I'm going to introduce Dr. Devaprasad Chattopadhyay now. He joined late because of some technical difficulties, but he is a very known name in the field of virology. He is specifically interested in discovering ethnomedical leads for curing difficult to treat diseases. He has established personal contacts with diverse ethnic communities of Andaman and West Bengal, with the objective of translating traditional information into innovations, nothing less than innovations. So far, Dr. Chattopadhyay is credited with 4 leads, 8 patents, 4
Hello.
books. Yes.
Hello,
Yes. Can you hear me?
I am Dr. Chattopadhyay speaking from Dr. Subhas Mondal phone
Oh, acha, acha.
because I am not able to get connected through my phone number. Okay.
Okay. Just now I'm introducing you. Can you hear me?
Yes, I'm just listening you. Tell.
Okay.
Asha kori. Thank you.
So,
চলে গেল কোথায়? Oh,
so
ঠিক আছে।
far, Dr. Chattopadhyay is credited with 4 leads, 8 patents, 4 books, and a mind-boggling 163 research articles in prestigious journals. He has produced 17 PhDs and completed several task force projects in ethnomedicine of marginalized communities. In April 2020, Dr. Chattopadhyay had set up a state-of-art COVID-19 testing laboratory with the help of Karnataka government. Currently, he is serving as the director at ICMR National Institute of Traditional Medicine, Belagavi, Karnataka. Now, I hand over the session to Dr. Chattopadhyay.
Thank you very much, Dr. Vijaya, for your brief introduction. First of all, you know that today's topic is repurposing drugs, particularly in case of COVID. Okay. And it is at par with the theme of this today's conference of NIHSM. Regarding repurposing of drugs, I will just briefly tell you what is repurposing and why repurposing is drug is required for COVID-19 disease. And hopefully you all know that COVID-19 is a viral disease. And this is a very new, unique virus, or you call it as an emerging zoonotic virus. The virus is first time detected in China. However, many of the people do not know what exactly the virus is. Virus is a life form, but it is an entity which challenges the very existence of living. You see, human evolution is indebted to the viruses, and our DNA contains the genetic code or genetic part of many viruses time to time. So we are co-evolved with the viruses, and virus has different beneficial side also. But with respect to COVID, this virus is unique because they are metabolically inert, acellular nucleoprotein particles, either consisting of DNA or RNA, that may be single-stranded or maybe double-stranded, maybe positive sense, maybe negative sense. So this is a very definition of virus, and particularly COVID virus. We first came to know about COVID virus in 2002-03 when a disease was reported called as severe acute respiratory syndrome caused by a special variety of virus, in which the virus which is called as coronavirus, because they have outermost glycoprotein spike, looks like the solar corona. And in 1960 onwards, 4 coronaviruses have been identified. Those viruses belonging to beta and alpha coronaviridae, they are responsible for mild cold and cough for human being. 15 to 20% of human population, or 15 to 20% of cold and cough and cold disease is caused by coronaviruses. But in 2002-03, first a virus is known that attack the upper respiratory tract infection and may go to the severe case also. That is called as COVID. and now it is called as COVID-1. Second COVID virus was reported from the Middle East called as MRSA, Middle East Respiratory Syndrome, in 2012-13, and lastly in 2019 at China. This virus is not man-made. You know, the distinct studies, a series of papers appeared in Nature and Proceedings of the National Academy of Science in last few months clearly indicate that a bat species contain more than 60 viruses, of which 12 belongs to coronavirus. And bat has a very peculiar immune system that cannot, and virus cannot able to multiply rapidly to cause a disease. So bat is called as reservoir, not only the host. Reservoir, host. This coronavirus probably interacting with other group of coronaviruses, alpha, beta, gamma, delta, and therefore genetically distinct from others. And interestingly enough, this is a single-stranded positive sense RNA coronavirus. That means this RNA virus or RNA act as messenger RNA. It does not go for the transcription, rather it can translate it into protein within the host cell without the help of host DNA. This is first uniqueness. Second uniqueness, RNA viruses are rapidly multiplying virus, and therefore when they are going to multiply rapidly, they can produce huge number of progeny virus, and there are several mistakes called as mutation. But here in this virus, there is a error-prone mechanism. But in this virus, there is a proofreader. So when the virus goes for multiplication, the mistake it has been done can be rectified, and that's why virus can able to survive. And so far, in the last 2 years, only 10 or 17 mutations takes place. It's a very alarming situation. The virus adapted in such a form which is found only in the higher forms. Second point. Third point, in 2020 April, my interaction with different media, mass media, TV, the Hindu, Daily Telegraph, then many other newspapers of India, as well as German Herald, I repeatedly told one thing: this is a virus came from animals, so initially it was animalistic in behavior. It lost its home, so it was very aggressive. And thirdly, when it's adapted to human being, then it will be slower and milder. And the days are coming. And as we are in a, you see, in a sea, you can find the waves, first wave, second wave, third wave. And in the vast number of human population like Janasamudra, you also find the third wave. But third wave may be not similar to second wave, because second wave is due to human failure also. So this is a very, very nutshell about the virus. Now, why repurposing is required? Because you know, viral, antiviral, which is supposed to inhibit or kill or destroy the virus, virus cannot be killed because virus is metabolically inert nucleoprotein particle. It has no cellular architecture. It has everything, but not the cooking system. That's why it goes to the human being, take the help of whole human system and multiply. Now what happened? If you target a virus which is made up of nucleoprotein and nucleic acid, the host DNA also contains protein and nucleic acid. So there is every chance the host DNA can be affected. And that's why all antivirals are toxic to the host. Secondly, almost 40 antivirals are available, and all antivirals are of nucleic acid derivative, either purine derivative or pyrimidine derivative. They have limitations. So new antivirals has not been generated in last 50 years. So it is very difficult to develop antivirals. That's why I think that it is better to target host virus interaction rather than virus itself. If you want to target the virus, you have to target very specific site of the virus so that host cannot be affected by the virus. Now there are a number of antiviral drugs and related drugs. The drugs are basically multifunctional. and a single drug act differently at different dose level. There are several examples. That's why some of the known drugs are used in this viral disease, so that if possible, if that drug can prevent this virus to kill the human being, that will be very beneficial. And that is called repurposing of known antivirals to this new virus. What are the benefits? Number one benefit, cost effective, because already the drug is used in certain other diseases. So toxicity and all other data is available. You don't have to go for any further study. Secondly, the dose variation. If it is lower dose, then there is no problem. If it is higher dose, then you have to study differently. Then mechanism of action or site of action or mode of action. So these are the few studies that can help to repurpose already known drug. And that's why in case of this virus, because this virus is known just 1.5 years, and in these 18 months, huge research throughout the world has taken place. And although a certain period of time was during pandemic time, it was locked down throughout the world. And probably this is the only virus that can show the human being the atomic power is not any power. A teeny ultramicroscopic metabolically inert nucleoprotein particle can create devastation in the superpower nations also. So you see, to nature, man has nothing to do. And this is a nature. This is a nature. Rather, you can say it is a natural disaster, not man-made disaster. Okay. So now repurposing help to identify a series of compounds that have got to some extent or some ability to manage this virus, to inhibit different parts of the virus. I can give you first example is chloroquine, which is a parasitic drug, antiparasitic drug. But here the drug helps in changing the pH pattern of the host and the infected cell. And some other, there are 4 or 5 points in which chloroquine can work. So people initially take that one. Second drug comes is remdesivir. Vir, the title vir or suffix vir means antivirals. This drug is RNA-dependent RNA polymerase inhibitor, which is available in RNA viruses. It was studied, it was developed for other viruses, but it was not very successful. Here people are using and getting to some extent results also. There are several other drugs also. I cannot go for naming one by one. If you are interested, you can go to the net and put the name, you will get a several paper, and I have 8 papers in this direction of a very prestigious journals like Phytomedicines, Phytotherapy Research, then American Society for Microbiology Journal, a number of journals you will find. In last 1.5 years, we have published 8-9 papers on this aspect, and we are developing a spray to prevent the spread of this virus through traditional medicine cocktail. So repurposing is basically use of other drugs to cut off the time and money if that drug can able to either collectively or biotically or in a combination can able to prevent the multiplication of the entry of the virus, multiplication of the virus, assembly of the virus, release of the virus. There are 6 steps actually in any viral infections. This is no exception. But this virus uses 2 different routes for entry, either through endocytosis or through attachment, effusion, and penetration. And in all the cases, not only one protein, there are 8 proteins of the host can help the virus to go inside. So any blocking of any of the protein, like ACE2, you all know, angiotensin renin system ka, ACE2, and that is related to our blood pressure. And that's why some ACE2 inhibitors are used for managing the blood volume. Okay, and other parameters also. So ACE2 is the primary site, but if the virus is not able to only use ACE2, when it attaches with the ACE2, and then it takes the help of transmembrane protein serine protease 2 as well as furin. These 2 proteins collectively help the virus not only to attach with the ACE2 firmly, but also go inside and undergoes multiplication. So these two proteins are very important, and there are a number of inhibitors of these two proteins. Many of the known drugs also work with it, and many plant compounds like luteolin, like ethanolides. There are several compounds that work against these two proteins. So it can be blocked by repurposing already known phytomolecules or already known drugs. Secondly, when the virus go inside, then the membrane protein PL-PRO, papin-like protease, that does two things. One, it prevents the interferon 1 secretion of interferon 1, which is helpful for stopping the virus or putting the virus away from the host interaction. But this PL-PRO of the virus stops the multiplication of DNA of the host, as well as it prevents the synthesis or secretion of interferon 1. So antiviral immunity by the host will stop. And then comes the main protein called as main protease, or 3-chymotrypsin-like protease and RNA-dependent RNA polymerase. So these 4 steps are very important for the virus. If we can collectively use the old already known drug in different part of the virus in a cocktail, which people are using, so the virus can be controlled. And that's why throughout the globe repurposing is still going on, and repurposing is a way to manage. You can say the new viruses, emerging viruses, or re-emerging viruses also. So over to the moderator.
Madam, you are on mute. Madam, you are on mute. Please unmute yourself.
Thank you very, very much for such wonderful talk. It was simple, and though I don't belong to the subject, I could also understand it. So I think the audience could follow. So to summarize, the 2 speakers have gone through 2 different aspects. Dr. Chattopadhyay has given us the basis. He said that there are 2 approaches. All antiviral drugs are toxic. So if we take one antiviral drug directly to interfere with the replication of the virus, it's definitely going to do some harm to the body. The second, the safer aspect is to target the host virus interactions. And he has given us the examples with 2 drugs. One was famous chloroquine, hydroxychloroquine, which was the controversy, which came into controversy because of US and India tussle for the supply of the drug. For some time, it was always in the headlines. He has given us the mechanism of action also. He said, this drug can increase the pH, and that controls the replication of the virus within the cell, if I'm not wrong. And the other drugs. Dr. Bose, before this, has also gone to the regulatory aspects. She said the background story, how Indian government has prepared to combat this new situation, the background work, the regulatory guidelines, then how actually we had fought. We had, being a huge population also, we are not doing very bad. We have successfully controlled some. There were some failures, but somehow with the effort, with the continuous effort, we have controlled it. So this was the, in very short, the gist of the talk. But I have some questions to both of you. So first to Debuda. I have noted it. You said all antivirals are toxic. Then what was the basis of selection of repurposed drugs?
Actually, it depends upon the which we are going to target. If our aim is to save the life somehow, then toxicity does not matter. Okay. Here the life is the primary target because thousands of people are dying throughout the world. So authority and government and all the states wanted to stop the killing of the human lives, and that's why toxicity is a secondary thing that can be manageable. But life, in the cost of life, toxicity cannot come, and that's why even all antivirals are toxic. We have to take the antivirals to save the life. So, out of all the antivirals available to date, acyclovir is called as the less toxic drug. Otherwise, all other antivirals are more toxic than acyclovir.
Okay. Now next to Dr. Bose. From your lecture I could get, the expenditure involved in drug repurposing is much less compared to discovering a new drug. COVID drugs are expensive. Don't you think companies are trying to make a quick buck on this? What is your take?
So when we, just previous to this, yes, when they're coming back with too much compounds, see, at one point of time, we had nothing. Like when the first drug came, favipiravir tablets, as a repurposed drug for the treatment of mild to moderate COVID-19, we had nothing actually. The vaccine was not there. There was nothing. So there was not a single drug in the therapeutic armamentarium, we can say. So we needed something that, yes, which will assure this population, yes, it is working. And actually, because it was already tested in influenza, and some of the countries like PMDA, or it was already approved in other countries and been started being used, even with a lot of toxicity, as sir was saying. They were saying that remdesivir with so much toxicity, and still remdesivir has been approved by US FDA for this particular treatment in the moderate to severe disease. So what was important is that we have to do this benefit risk assessment, and we have to bring confidence, build in confidence that how the country will fight this. When we know that vaccine will take it is good that the vaccine is working. At least it is working. At least there is some antibodies produced, which is actually reducing this infection. But at one point of time, we had nothing in our hand. Yes, there may be now, once one manufacturer came, there are subsequent, because India is a generic manufacturing country. So there are so many manufacturers coming with different populations. That's normal. That's normal. Anybody who is a manufacturer, they will try to take advantage out of this. But yes, we are actually working in this manner in India. As we have many manufacturers, we know that the drug price gets so reduced to a sustainable amount. We just give a permission, and then we have another, and we have second, third, and so many, and so on, and that the way the price gets reduced. Yes, everybody tries to manufacture, but that is the reason we have been called as the pharmacy of the world, that our manufacturers come up so first. And yes, and everybody trying to convert the monoclonal antibodies into a COVID drug. Yes, so that assessment also is being done by the subject expert committee, by the regulators, to ensure that any unnecessary compounds doesn't come into the treatment mode. So I think,
Yes, the price is reduced. Some of the companies have reduced the price as much as 2,000 rupees also.
and
so that was under pressure.
Yes, true.
And to Dr. Chattopadhyay, once again, you said something about the development of a spray. Can you elaborate a little more?
From our childhood, particularly the people and not in touch of very high sophisticated family city, our grandmas really run the home, run the soul of the home. In any type of infection, particularly upper respiratory or lower respiratory tract infection, congestion in the chest, in season change during summer to winter or rainy season, something happens, even in the bad stomach, they used to give some plant recipe in the form of say boiling কি বলে ওটাকে ওই বলো না আরক টাইপের করে দিত আর কি হ্যাঁ তো that we used to take. Even you see everybody understand about the basak না অ্যাডাটোডা ভাসিকা হ্যাঁ তো খাবি না মানে তোর ঘাড় খাবে ধরে খাওয়া তো মধু দিয়ে মনে আছে তো my grandmother was a person who used to give this type of medications or medicaments to a number of people in the villages. Some got good result, some may not, and in sometimes some side effect also comes. So that I have seen, and I was the supplier of the plants from the nearby forest. So it was in my blood, you can say. When I got the opportunity to come back from England, London, and joined in a ICMR, particularly RMRC Port Blair. So I thought that I will work with the traditional system of medicine. And you know, I'm the first person in India who started the scientific study of tribal ethnomedicine practices, particularly the validation of tribal ethnomedicine through the science. And we have published, JBC, we have published almost all very famous biomedical journal. And that is our pride also. You see, I'm the first person from country who has maximum publication on antivirals from traditional medicine. Whatever virologist exists in India, nobody can supersede my number of publications till date. Okay, so my experience in this particular field, I have prepared on the basis of my grandmother recipe and some traditional healers recipe, I combine together and prepare a formula. Then I go to study the ancient literature and then contemporary literature, the biological activity of these plant, of those extracts. And I have identified few such plants having little bit antiviral activity, anti-inflammatory activity, and immunomodulatory activity. Collectively, we form a combination of 7 plants. And then I go to take the help of bioinformatics study with molecular docking studies. We try to find out which plant and which compound of the plant is interacting with the viral spike protein, membrane protein, then nucleoprotein. Okay. So by that way, we formed a compound, a complex, and then we designed 3-4 tests. The first test gives us, we have taken the viral spike protein. Then we put the virus spike protein in 96-way plate, then incubate. Then we add the human ACE2 protein and see the binding. When we put our formula, the binding is prevented by 98.89%. And it has 10 times repeated. Then go for Western blot and other studies. Now we are preparing the pseudovirus particle and trying to see how our formula works. And third test will be the hamster test, in which we will infect the hamster and then subsequently see whether it can prevent, whether it can interact at a time, or whether it interact after the infection. Okay. So the study is going on, and hopefully another few months, 3-4 months, something will come out as a formulation. And being my touch with pharmacy department so long, I have a maximum pharmacy friend, and those friends are responsible for this idea as well as implementation of the idea. And I'm fortunate to have such type of elaborate career and elaborate knowledgeable scientist of pharmacy department of Jadavpur University. And hopefully we will be successful in this.
We are almost at the end of our session. Thank you, Dr. Chattopadhyay. Thank you very, very much. And Dr. Bose. You were so busy, still were kind enough to give us your precious time and gave that wonderful illuminating talk. Thank you once again for your time and energy.
Thanks a lot, Vijaya. Thanks a lot. After a
Thank
long
you.
time, I've great.
I enjoyed the session. I hope you too also.
Yes, of course, of course.
Thank you,
Okay,
Vijay.
okay,
Thank you.
okay, okay. Bye. দেখো বন্ধ সুভাষ বাঁচিয়ে দিল না তো প্রেস্টিজ চলো সুভাষ আমরা একটু নিচে দেখি কোনো দোকানপত্র পাই কিনা
না এই ওদের নিয়ে আসবে সিগারেট
বুঝছো
একটু
পাক্কা
থাকো
কিন্তু পেট খেটে গেল
টেস্টে চাও তারপরে
I would like to request Dr. Devprasad Chattopadhyay, sir. Can you please put your microphone on mute?
কি বলছে
Sir, you are audible. Can you please put your microphone on mute?
চলো অনেক হেল্প চলো
Thank you, Dr. Devaprasad Chattopadhyay and Dr. Robina Bose for your valuable insights. Thank you, Vijaya Madam, for handling and moderating the session with expertise. The next session is for cultural events. And for that session, I would like to hand the session over to Dr. Amrita Chakraborty. Thank you very much.
Thank you, everyone. Now we will proceed with the cultural event for the evening. And thank you, all the panelists, all the keynote speakers, and all the participants for being a part of our international conference. We will wait for a short while and we will be starting our cultural program within a time span of 5 to 10 minutes. Please be patient to witness the cultural extravaganza from the students of NSHM Knowledge Campus. Good evening, everyone. I think all of us are ready by this time to witness a bit different program from which food has been circulated from the morning. After all this brainstorming session, we now would like all of you to move on to a lighter note. Please enjoy the cultural program from the Pharmaceutical Technology Portal. The program has been compiled by our students and we hope that you will enjoy it. Now, let me introduce the anchors for the evening, our student, Mr. Anubhav Mondol, Miss Talha Ahmed, and Mr. Schroff Dash. Over to Anubhav, Talha, and Schroff. We all know India is a country of unity among diversity, and this diverse geographical, social, and cultural background makes India unique and beautiful. Nature has adorned India with high mountain ranges, vast seas, countless rivers and streams, seamless greeneries, dark forests, and sandy deserts, almost a mini world. India has an exceptional range of human strains which wonderfully amalgate to create a unique cultural identity. The mesmerizing art form from different states of India gives us a colorful bouquet of cultural heritage. Each state of India is blessed with a unique art form which differs considerably from that of its neighbor.
This evening, we would like to take you to a short trip to this distinct heritage. Made of 7 states known as the Seven Sisters, Northeast India have the potential to come out on top as the most interesting destination. It is a land alternated by hills and plains with a luxurious green cover. Strangely, each of the 7 states retain its own cultural identity. Let us witness the beauty of Northeastern culture via a dance performance. Titi Noshkar will dance to the tune of Bihure Lobo, a song to celebrate the harvest season.
Hello everyone, I am Somadeep Sarkar from 2008-12 batch Pharmacy Portal. Right now I am working as a area sales manager in India Metronic. I take care of the eastern part of our region.
My name is Ankita Sarkar and I am an alumni of NSHM College of Pharmaceutical Technology, Kolkata. I have done my B.Pharm from NSHM in the year of 2013 to 2017, working as an assistant professor at Adamas University.
My name is Sujay Pathak. I have done my B.Pharm and M.Pharm both from NSHM. I am really blessed that I am a NSHMite. So right now I am working for Cognizant Technology Solutions as a drug safety analyst.
I am Momita Haldar. I completed B.Pharm from NSHM Knowledge Campus, Kolkata in 2016. After completing B.Pharm, I joined Naipar Mohali to pursue MS Pharm in medicinal chemistry. Now I am enrolled at IIT Ropar at Department of Chemistry to pursue PhD.
I'm extremely sorry for the technical glitch. There is an issue with the platform, I think. Let us wait for a few while. I assure you that we will be again resuming back within a short while. Okay.
We all know India is a country of unity among diversity, and this diverse geographical, social, and cultural background makes India unique and beautiful. Nature has adorned India with high mountain ranges, vast seas, countless rivers and streams, seamless greeneries, dark forests, and sandy deserts. Almost a unique cultural identity. The mesmerizing art formed from different states of India gives us a colorful bouquet of cultural heritage. Each state of India is blessed with their unique art forms, which differs considerably from that of its neighbor.
This evening, we would like to take you to a short trip to this distinct heritage. Made of 7 states known as the Seven Sisters, Northeast India have the potential to come out on top as the most interesting destination. It is a land alternated by hills and plains with a luxurious green cover. Strangely, each of the 7 states retains its own cultural identity. Let us witness the beauty of Northeastern culture via a dance performance. Harvest season. We are really sorry. There was a technical glitch. We will restart the program again. So please stay with us.
We all know India is a country of unity among diversity, and this diverse geographical, social, and cultural background makes India unique and beautiful. Nature has adorned India with high mountain ranges, vast seas, countless rivers and streams, seamless greeneries, dark forests, and sandy deserts, almost a mini world. India has an exceptional range of human strains which wonderfully amalgate to create a unique cultural identity. The mesmerizing art forms from different states of India gives us a colorful bouquet of cultural heritage. Each state of India is blessed with their unique art forms which differ considerably from that of its neighbor.
This evening, we would like to take you to a short trip to the distinct heritage. Made of 7 states, known as the Seven Sisters, Northeast India have the potential to come out on top as the most interesting destination. It is a land alternated by hills and plains with a luxurious green cover. Strangely, each of the 7 states retain its own cultural identity. Let us witness the beauty of Northeastern culture via a dance performance. Tithi Noshkar will now dance to the tune of Bihure Lago, a song to celebrate the harvest season.
বিহুরে লগন মধুরে লগন আকাশে বাতাসে লাগিল রে চম্পা ফুটিছে চামেলি ফুটিছে তার সুবাসে ময়নায় আমার ভাসিল রে বিহুরে লগন মধুরে লগন আকাশে বাতাসে লাগিল রে চম্পা ফুটিছে চামেলি ফুটিছে তার সুবাসে ময়নায় আমার ভাসিল রে হলুদ বরণ মেখলায় তার যৌবন উছলায় লাল ওড়নার আড়াল দিয়া চক্ষু দুটি চায় হলুদ বরণ মেখলায় তার যৌবন উছলায় লাল ওড়নার আড়াল দিয়া চক্ষু দুটি চায় খোঁপায় টগর ময়না বুঝি আমায় খুঁজে আয় বসন্তে এই বিহুর লগন উত্তল হয়ে যায় বিহুরে লগন মধুরে লগন আকাশে বাতাসে লাগিল রে নাচিতে নাচিতে আর ভরা যৌবন বিহুর সাজে সবার মাঝে আসিল রে তাহার নাচ দেখি আমার অঙ্গ অবস্থায় ঢোলের কাঠি দুহাত থেকে আপনি খসি যায় তাহার নাচ দেখি আমার অঙ্গ অবস্থায় ঢোলের কাঠি দুহাত থেকে আপনি খসি যায় জগৎ জুড়ি বিহুর পরব খুশি যে ছড়ায় বসন্তে এই বিহুর লগন উত্তল হয়ে যায় বিহুরে লগন মধুরে লগন আকাশে বাতাসে লাগিল রে চম্পা ফুটিছে চামেলি ফুটিছে তার সুবাসে ময়নায় আমার বাসিল রে বিহুরে লগন
for such beautiful performance.
The vibrant state of Gujarat has significant contribution to the culture of India. The state contributes a lively array of art, architecture, and culture, all of which are unique in its own sense. Welcome to the colorful and of Gujarat, and let's enjoy a Gujarati dance performance by Dipanvita Naskar.
जी 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 गोरी के कंगना आज छन छनछनाक पायल संग बाजे सर पर चूनर ओढ़े निकले की आज राधिक लहरा लहरा के गोपियों संग और वो गगन झूमेंगे संग संग सब पे चढ़ेगा आज प्रेम रंग रंगी गुलाल होगा सोचो क्या हाल होगा नाचेंगे प्रेम रोगी धम धम धातिला धिदकिट ढिला बाजे मृदंग धना धन बाजे छम 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 चमाक झांझर झम झम चमाक घुंघरू गम धम गमाक चमक चम चमाके बाजे रे बाजे रे बाजे रे बाजे रे ढोल बाजे बाजे रे बाजे रे बाजे रे ढोली तारो ढोल बाजे ढोल बाजे ढोल बाजे ढोल के धम धम बाजे ढोल ढोली तारो ढोल बाजे ढोल बाजे ढोल बाजे ढोल जो धम धम बाजे ढोल हे छोरी बड़ी अनमोल मीठे मीठे इसके बोल आंखें इसकी गोल 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 तो ढम ढम बाजे ढोल हाँ हाँ छोरा है नटखट बोली है पटपट अरे छेड़े मुझे बोले ऐसे बोल तो ढम ढम बाजे ढोल हे बाजे रे बाजे रे बाजे रे धम धम ढोल बाजे ढोल बाजे ढोल बाजे ढोल तो धम धम बाजे ढोल तो धम 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 बाजे ढोल
Thank you, Dipanvita, for presenting a beautiful dance.
Leaving Gujarat, now let's move towards Rajasthan, the land of the heroic kings and beautiful queens. Full of deserts and splendid forts, this state is one of the hottest parts of our country. Rajasthani culture is replete with glorious history, which is reflected in its spirited dance performance and mesmerizing music. Let's visit the state with a dance performance by Geeta Shah.
ढोल
Thank you, Titash, for your entertaining performance.
Leaving the north, let's enter South India. The vast South Indian plains has so many things to offer. Unique architectural designs that sing stories of colorful past, mesmerizing Kathakali dance and captivating Kannada music. Experience the richness of this culture with a traditional dance performance by Shikriti Paul.
जटाटवी गलज्जल प्रवाह पावितस्थले कलेवलम्ब्य लम्बितां भुजङ्ग तुङ्गमालिकान्तमड्डमड्डमन्निनादवड्डमर्वयं चकार चण्डताण्डवं तनोतु नः शिव शिवम् जटाभुजंग पिंगल स्फुरत्फणामणि प्रभा कदम्ब कुङ्कुमद्रव प्रलिप्त दिग्वधूमुखे मदान्ध सिन्धुर स्फुरत्वगुत्तरीय मेदुरे मनो विनोदमद्भुतं बिभर्तु भूत भर्तरि ॐ नमः शिवाय सदा शिवं भजाम्यहं सदा शिवं भजाम्यहम् ॐ नमः शिवाय प्रफुल्ल नील पंकज प्रपंच कालिमप्रभावलम्बिकण्ठ कन्दली रुचि प्रबद्ध कन्धरं स्मरच्छिदं पुरच्छिदं भवच्छिदं मखच्छिदं गजच्छिदान्धकच्छिदं तमन्तकच्छिदं भजे अखर्व सर्व मंगला कला कदम्ब मंजरी रस प्रवाह माधुरी विजृंभणा मधुव्रतं स्मरान्तकं पुरान्तकं भवान्तकं मखान्तकं गजान्तकान्धकान्तकं तमन्तकान्तकं भजे इदं हि नित्यमेव मुक्तमुत्तमोत्तमं स्तवं पठन् स्मरन् ब्रुवन् नरो विशुद्धिमेति सन्ततं हरे गुरौ सुभक्तिमाशु याति नान्यथा गतिं विमोहनं हि देहिनां सुशङ्करस्य चिन्ता विमोहनं हि देहिनां सुशङ्करस्य चिन्ता विमोहनं हि देहिनां सुशङ्करस्य चिन्ता
Thank you, Shikriti, for your lovely performance.
Known as the cultural capital of India, West Bengal stands out as a rare orchid in our cultural bouquet. Blessed with a landscape full of hills, plains, and a sea, Bengal is perhaps the most diverse part of India in terms of topography. Bengal is also the home of NSHM Knowledge Campus, which is hosting this conference. We welcome you to a glimpse of its cultural beauty.
দুই বিঘে জমি, কবিগুরু রবীন্দ্রনাথ ঠাকুর। শুধু বিঘে দুই ছিল মোর ভুঁই, আর সবই গেছে ঋণে। বাবু কহিলেন, বুঝেছ উপেন, এ জমি লইব কিনে। কহিলাম আমি, তুমি ভূস্বামী, ভূমির অন্ত নাই। চেয়ে দেখো মোর আছে বড় জোর, মরিবার মতো ঠাঁই। শুনে রাজা কহে, বাপু জানো তো হে, করেছি বাগানখানা। পেলে 2 বিঘে প্রস্থে ও দিঘে সমান হইবে টানা, ওটা দিতে হবে। কহিলাম তবে, বক্ষে জুড়িয়া পানি, সজল চক্ষে করুণ রক্ষে গরিবের ভিটে খানি। সপ্ত পুরুষ যেথায় মানুষ, সে মাটি সোনার বাড়া। দৈনের দায়ে বেচি বসে মায়ে, এমনই লক্ষ্মী ছাড়া। আঁখি করি লাল, রাজা ক্ষণকাল রহিলা মৌন ভাবে। কহিলেন শেষে ক্রূর হাসি হেসে, আচ্ছা সে দেখা যাবে। পরে মাস দেড়ে ভিটে মাটি ছেড়ে বাহির হইন পথে। করিল ডিক্রি সকলে বিক্রি, মিথ্যা দেনার ক্ষতে। এ জগতে হায় সেই বেশি চায়, আছে যার ভুরি ভুরি। রাজার হস্ত করে সমস্ত, কাঙালের ধন চুরি। মনে ভাবিলাম, মোরে ভগবান রাখিবে না মোহ গর্তে। তাই লিখে দিল বিশ্ব নিখিল, 2 বিঘার পরিবর্তে। সন্ন্যাসী বেশে ফিরি দেশে দেশে, হইয়া সাধুর শিষ্য। কত হেরিলাম মনোহর ধাম, কত মনোরম দৃশ্য। ভূধরে সাগরে বিজনে নগরে, যখন যেখানে ভ্রমি। তবু নিশিদিন ভুলিতে পারিনে, সেই 2 বিঘে জমি। হাটে মাঠে বাটে এই মতো কাটে বছর 15-16। একদিন শেষে ফিরিবারে দেশে, বড়ই বাসনা হলো। নম 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 সুন্দরী মম জননী বঙ্গভূমি। গঙ্গার তীর স্নিগ্ধ সমীর, জীবন জুড়ালে তুমি। অবারিত মাঠ গগন ললাট, চুমে তার পদধূলি। ছায়াশু নিবিড় শান্তির নীর, ছোট ছোট গ্রামগুলি। পল্লব ঘন আম্র কানন, রাখালের খেলা গেহ। স্তব্ধ অতল দিঘি কালো জল, নিশিত শীতল স্নেহ। বুক ভরা মধু বঙ্গের বধূ, জল লয়ে যায় ঘরে। মা বলিতে প্রাণ করে আনচান, চোখে আসে জল ভরে। 2 দিন পরে দ্বিতীয় প্রহরে প্রবেশিনু নিজ গ্রামে। কুমোরের বাড়ি দক্ষিণে ছাড়ি, রথ তলা করি বামে। রাখি হাট খোলা, নন্দির গোলা, মন্দির করি পাছে। ত্রিশাতুর শেষে পহিছিনু এসে, আমার বাড়ির কাছে। ধিক ধিক ওরে শত ধিক, নীলাজ কুলটা ভূমি। যখনই যাহার তখনই তাহার, এ কি জননী তুমি। সে কি মনে হবে একদিন যবে ছিলে দরিদ্র মাতা। আঁচল ভরিয়া রাখিতে ধরিয়া, ফল ফুল শাক
পাতা। আজ কোন রেতে কাকে ভুলাইতে ধরেছ বিলাস বেশ, পাঁচরঙা পাতা অঞ্চলে গাঁথা পুষ্পে খচিত কেশ। আমি তোর লাগি ফিরেছি বিবাগী, গৃহ হারা সুখহীন। তুই হেথা বসি ওরে রাক্ষসী, হাসিয়া কাটাস দিন। ধনির আদরে গরব না ধরে এতই হয়েছ ভিন্ন, কোনখানে লেশ নাহি অবশেষ সেদিনের কোনো চিহ্ন। কল্যাণময়ী ছিলে তুমি ওই ক্ষুধা হারা সুধা রাশি, যত হাসো আজ যত করো সাজ, ছিলে দেবী হলে দাসী। বিদীর্ণ হিয়া ফিরিয়া ফিরিয়া চারিদিকে চেয়ে দেখি, প্রাচীরের কাছে এখনো যে আছে সেই আম গাছ। একই বসে তার তলে নয়নের জলে শান্ত হইল ব্যথা। একে একে মনে উদিল স্মরণে বালক কালের কথা। সেই মনে পড়ে জ্যৈষ্ঠের ঝড়ে রাত্রে নাহিকো ঘুম, অতি ভোরে উঠে তাড়াতাড়ি ছুটি আম কুড়াবার ধুম। সেই সুমধুর স্তব্ধ দুপুর পাঠশালা পলায়ন, মনে ভাবিলাম হায় আর কি কোথায় ফিরে পাবো সে জীবন। সহসা বাতাস ফেলি গেল শ্বাস শাখা দুলাইয়া গাছে, দুটি পাকা ফল লোভিল ভূতল আমার কোলের কাছে। ভাবিলাম মনে বুঝি এতক্ষণে আমারে চিনিল মাতা, স্নেহের সে দানে বহু সম্মানে বারেক ঠেকানো মাথা। হেনকালে হায় জমদূত প্রায় কোথা হতে এলো মালি, ঝুটি বাঁধা উড়ে সপ্তম সুরে পাড়িতে লাগিল গালি। কহিলাম আমি তো নীরবে দিয়েছি আমার সব, শুধু দুটি পাকা ফল করি তার অধিকার, এত তারি কলরব। চিনিল না মোরে নিয়ে গেল ধরে কাঁধে তুলি লাঠি গাছ। বাবু ছিপ হাতে পারিষদ সাথে ধরিতেছিলেন মাছ। শুনি বিবরণ ক্রোধে তিনি কন মারিয়া করিব খুন। বাবু যত বলে পারিষদ দলে বলে তার শতগুণ। কহিলাম আমি শুধু দুটি পাকা আম ভিখ মাগি মহাশয়। বাবু কহে হেসে বেটা সাধু বেশে পাকা চোর অতিশয়। আমি শুনে হাসি আখি জলে ভাসি, এই ছিল মোর ঘটে। তুমি মহারাজ সাধু হলে আজ, আমি আজ চোর বটে।
Thank you, Shayanika, for presenting such a beautiful performance.
So far, we have presented the individual flowers, bunches of saffron, blue, and green, gladioli. In the same garden, they make a feast for the eye. It creates a wonderful cultural ambience that teaches us to practice all the great virtues of human existence: equality, freedom, and secular thought. Please enjoy the last performance of the cultural evening with the dance Desh Rangila Rangila by Tithi and Dash.
Thank you everyone for being patient and witnessing the cultural extravaganza on behalf of the students of NSHM Knowledge Campus. I would like to thank all the participants who made this cultural event successful. I would also like to thank Debar Hobor and Maulik Mondol for providing technical support, which made this cultural program smooth as well as organized. Last but not the least, thank you all the members of the cultural committee of ENACON PH 2021 for helping and supporting us. There was a technical glitch in the beginning, and I am very happy that all of you waited patiently with us and you enjoyed the performance. Now, let's move on again to the home of the conference and witness the magic of Rabindranath Tagore by a song performed by Shaktopriyo Guru, one of our students.
যদি তোর ডাক শুনে কেউ না আসে, তবে একলা চলো রে, তবে একলা চলো, একলা চলো, একলা চলো, একলা চলো রে। যদি তোর ডাক শুনে কেউ না আসে, তবে একলা চলো রে। যদি কেউ কথা না কয়, ওরে ওরে ও অভাগা, কেউ কথা না কয়, যদি সবাই থাকে মুখ ফিরায়, সবাই করে ভয়, যদি সবাই থাকে মুখ ফিরায়, সবাই করে ভয়, তবে পরাণ খুলে ও তুই মুখ ফুটে তোর মনের কথা একলা বলো রে। যদি তোর ডাক শুনে কেউ না আসে, তবে একলা চলো রে। যদি সবাই ফিরে যায়, ওরে ওরে ও অভাগা, সবাই ফিরে যায়, যদি গহন পথে যাবার কালে কেউ ফিরে না চায়, যদি গহন পথে যাবার কালে কেউ ফিরে না চায়, তবে পথের কাঁটা ও তুই রক্ত মাখা চরণ তলে একলা দলরে। যদি তোর ডাক শুনে কেউ না আসে, তবে একলা চলো রে। যদি আলো না ধরে, ওরে ওরে ও অভাগা, আলো না ধরে, যদি ঝড় বাদলে আধার রাতে দুয়ার দেয় ঘরে, যদি ঝড় বাদলে আধার রাতে দুয়ার দেয় ঘরে, তবে বজ্রানলে আপন বুকের পাঁজর জ্বালিয়ে নিয়ে একলা চলো রে। যদি তোর ডাক শুনে কেউ না আসে, তবে একলা চলো রে, তবে একলা চলো একলা চলো একলা চলো একলা চলো রে, যদি তোর ডাক শুনে কেউ না আসে, তবে একলা চলো রে।
Thank you, Schroff, for such a wonderful performance. It's time for witnessing the magic of Rabindranath Tagore once again by a beautiful dance performance by one of our students, Miss Sharika Kosham. I'm sorry for another technical glitch. We will now witness the magic of Kaji Nazrul Islam by a dance performance of Tithi Noshkar.
সৃজন ছন্দে আনন্দে নাচন রাজে, মহাকাল প্রলয় তাল বলো বলো, সৃজন ছন্দে আনন্দে নাচন রাজে, মহাকাল প্রলয় তাল বলো বলো, সৃজন ছন্দে। ছড়াক তব জটিল জটা, শিশু শশীর কিরণ ছটা, ছড়াক তব জটিল জটা, শিশু শশীর কিরণ ছটা, উমারে বুকে ধরিয়া সুখে, উমারে বুকে ধরিয়া সুখে, দোল দোল সৃজন ছন্দে আনন্দে নাচো নটরাজে, মহাকাল প্রলয় কাল বলো বলো, সৃজন ছন্দে। শ্রোতা মন্দাকিনী সুরধুনী তরঙ্গিনী,
At NSHM, we have Angan, Center of Creative and Performing Arts. Here, students are trained and given a platform for showcasing their hidden talents. Now, let us witness the performance of the students of Angan, NSHM Center of Creative and Performing Arts.
যদি জানতে আমার কিসের ব্যথা তোমায় জানাতাম, যদি জানতে কে যে আমায় কাঁদায়, কে যে আমায় কাঁদায়, আমি কি জানি তার নাম, যদি জানতে আমার কিসের ব্যথা তোমায় জানাতাম, যদি জানতে কোথায় যে হাত বাড়াই নিচে ফিরি আমি কার পিছে, কোথায় যে হাত বাড়াই নিচে ফিরি আমি কার পিছে, সব যেন মোর বিকিয়েছে পাইনি তার দাম, যদি জানতে আমার কিসের ব্যথা তোমায় জানাতাম, যদি জানতে এই বেদনার ধ্বংসে কোথায় ভাবি জনম ধরে, ভুবন ভরে আছে যেন পাইনি জীবন ভরে, এই বেদনার ধ্বংসে কোথায় ভাবি জনম ধরে, ভুবন ভরে আছে যেন পাইনি জীবন ভরে, সুখ যারে কয় সকল জনে বাজাই তারে ক্ষণে ক্ষণে, সুখ যারে কয় সকল জনে বাজাই তারে ক্ষণে ক্ষণে, গভীর সুরে চাইনি চাইনি, গভীর সুরে চাইনি চাইনি বাজে অবিশ্রাম, যদি জানতে আমার কিসের ব্যথা তোমায় জানাতাম, যদি आजा रे मेरे पिया, हो मेरे पिया, अब आजा रे मेरे पिया। तेरी हवा में बहने लगा हूँ, जाने मैं कैसे तेरा हुआ हूँ। मुझे तो लगता है मैं शायद तेरे दिल की दुआ हूँ। हाँ, तुझको जो पाया तो जीना आया। अब ये बसर है, अब तुझे भी जाना किधर है। जहाँ रहे तू मैं वो जहाँ हूँ, जिस ये दिन तू मैं वो समा हूँ। तेरी वजह से नया नया हूँ, पहले कहा ना मैंने अब ये तुमसे कहने लगा तुझको जीना গো ভারত লক্ষ্মী উঠো আজি জগত জনপূজা দুঃখ দৈন্য সবনাশী কর দূরিত ভারত লজ্জা ছাড়ো গো ছাড়ো শোক সজ্জা কর সজ্জা পুন কমল কনক ধন ধান্যে জননী গোলহ তুলে বক্ষে শান্তন বাস দেহ তুলে চোখে কাঁদিছে তব চরণ তলে ত্রিংশতি কোটি নর নারী গো কাণ্ডারি নাহি কো কমলা দুঃখ লাঞ্ছিত ভারত বর্ষে শঙ্কিত মোরা সব যাত্রী কালো সাগর কম্পন দর্শে তোমার অভয় পদ স্পর্শে নবহর্ষে পুন চলিবে তারণী শুভ লক্ষ্যে জননী গোলহ তুলে বক্ষে শান্তন বাস দেহ তুলে চোখে কাঁদিছে তব চরণ তলে ত্রিংশতি কোটি নর নারী গো
Thank you everyone for being a part of ENATCON PH 2021. I, Dr. Amrita Chakraborty, on behalf of NSHM Knowledge Campus, Kolkata, wish you all. I hope that all the participants, the speakers, the panelists, as well as the moderators have enjoyed the sessions and have gained some good insights. Now, let us end today's program with a dance performance by Shaimika Goswami. I hope that all of you will join us again back on tomorrow, sharp at 11 p.m. Thank you very much. Good night. Namaste.
একই সাজে গর্ব সব টুটিয়া মূর্ছি পরে লুটিয়া গর্ব সব টুটিয়া মূর্ছি পরে লুটিয়া সকল মম দেহ মন বিনা সমবাজে মহারাজ একই সাজে এলে হৃদয় পুরো মাঝে চরণ তলে কোটি শশী সূর্য মরে লাজে মহারাজ একই সাজে একই পুলক বেদনা বহিছে মধুবায়ে কাননে যত পুষ্প ছিল মিলিল তব পায়ে একই পুলক বেদনা বহিছে মধুবায়ে কাননে যত পুষ্প ছিল মিলিল তব পায়ে পলক নাহি নয়নে হেরি না কিছু ভুবনে পলক নাহি নয়নে হেরি না কিছু ভুবনে নিরখি শুধু অন্তরে সুন্দর বিরাজে মহারাজ একই সাজে এলে হৃদয় পুরো মাঝে চরণ তলে কোটি শশী সূর্য